Check.
You have seen the printed book? I've seen some publications on videos also. We have paleontology in it also.
It's important for now. Also knows about.
Yeah. And the question from Tamil Nadu.
Also taxonomist?
No. Economic monopoly.
Economic.
The Ranjit on the he was from the control of monopoly at higher identity. Tunisia is a very dynamic methodology from care of
Dynamic in the sense?
trade oriented. The ladies in field
Taking out the beneficial one and remove the parasitic one.
is doing very good work and very critical reviewer of our ministry. I appreciate that. And I give her a lot of job for that.
When you are active, more quality you get.
Dr. Rajan?
Yeah. Then you are better.
Dr. Rajan Sualian at Jammu. Only nematologist in
This is first time.
just for all managers, they were all ignorant. वहाँ पे कोई नहीं जानता था 8,000 साल पहले।
हेलो चेक 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 चेक
ये प्लान नेमाटोड्स का सिम्टम जब तक ऊपर नहीं बोलता है तब तक नहीं फॉर्मल बोलर नहीं करता है। ये प्रॉब्लम ये है, रूट नहीं देखता है कोई, ऊपर देखते हैं प्लान सपोर्ट करता है पर तो डायग्नोस्टिक सिम्टम नहीं होता है। इसलिए अभी से नहीं अभी थोड़ा कंसर्न हो गया है, अवेयरनेस भी काफी आ गई। हॉर्टिकल्चर क्रॉप अभी शुरू किया था हमने, मैं इंसिस्ट करता था पॉलिसी मेकर्स को कंपल्सरी करिए अपनी सिंपोजिया पॉलिसी मेकर्स का होना जरूरी है। मैंने हर मीटिंग में से हमारे
আমরা কি আসেনি সুসুনাকে ফোন করতে বলেছে তাই বলে
I think as a small group, we know each other.
We know each other very well.
Kind of family.
Devanand Das is a very good nematologist.
বলছি সুসুনা আসেনি এখনো সুসুনাকে ফোন করো সুসুনা আসেনি এখনো অডিও ভিজুয়ালে কেউ আসেনি আমার কাছে নাম্বার মেরে দিচ্ছি আমার নাম্বার আছে
कोशिश किया है तो काफी ट्रेंडिंग किया है तो वो काम आती है हमारे एडवाइजर्स ने हमें बहुत सिखाया। वही कोशिश की थी और आपके सामने सेकंड की वापसी है।
पेमेंट अभी सर कर रहा है तो सर हाँ वो होने वाला है साइड में चला जाएगा। कितना पड़ता है आपको?
अच्छा हाँ
मैं सेकंड की वापसी है कल देखते हैं।
अभी आए সুশান্ত সামনে আছে আমার সুশান্ত ওদিকে যাচ্ছে ওদিকে বেরিয়ে যাচ্ছে ঠিক আছে
चलिए अच्छा है, देखता हूँ कल कल कल।
हाँ बिल्कुल ठीक। हाँ हाँ
हेलो 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 ठीक हेलो हेलो ठीक हेलो 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 बाद में फोन कर, काय बोल। हाँ बोल। नहीं नहीं अब नहीं। हेलो हेलो
तो हम तो रिटायर्ड एक्टिव है मेरे
हेलो हेलो चेक हेलो 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 Hello, check. Hello. 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 Hello, check. Hello. Hello.
अगली तैयार करना जो है, तो खत्म हो जाता है जहाँ शुरू करते हैं वही तो होता है। हिसार में रहे they must take Sanders. नहीं बट है कि आप मेन स्ट्रीम में आइए ना, मेन स्ट्रीम में आके ही पता लगता है बहुत तो करते हैं। रहता है ट्रेडिशनल और एग्रीकल्चर वाले ना, वो फर्क रहता है थोड़ा सा शुरू से ही है वो, लेकिन हम लोग कोशिश करते हैं कि मैंने भी ट्रेडिशनल से ही शुरू किया था। मैं चंडीगढ़ में पैरासाइटोलॉजी से ही शुरू किया था, जूलॉजी से, डॉक्टर एन.के. गुप्ता के साथ। मैं तो पीएचडी में इधर आ गए, फिर अप्लाइड शुरू
यानी ये सुई जो बोले ना
অভিষেক কটায় শুরু হবে আমি ওই জন্যই কটায় শুরু হয় জিজ্ঞেস করেছি। আমি পরে আমার খেয়াল হলো, আপনাকে দেখে মনে হলো আমি
अब तो 10 बज रहा है ना। यहाँ पे आ जाओ। सर को लाइन में बाद में। हाँ नहीं, गाड़ी तो तुम्हारा बस है ना, रहेगा। गाड़ी कहाँ पे लगाएंगे सर? ये कॉन्फ्रेंस में सामने आ जाओ। हाँ हाँ हाँ, जहाँ तक आ रहा है वो लेके आओ, ठीक है। गेस्ट हाउस से आ रहा है।
কোথায় ও সই করে গেছি আমি তো ভালো সই করি না, তো বিমান খুলিনি, ভালো চলে এসছে। তোমাকে বলি ওর ডিমান্ডটা খুলুক। ডিমান্ডটা খুলবি, আমি ভালো আছি, চিন্তা করে সাহায্য করতে হবে না। এখান থেকে হবে না,
शाम को टाइम में फिर यहाँ से जाना
কেমন আছেন?
Hey.
গুড মর্নিং। আমার এটি বলতে গুড মর্নিং। আমি সংঘমিত্রা বন্দ্যোপাধ্যায়, ডাইরেক্টর, head of the Department of Division of Nematology. Please.
And you know, we crossed part 1 year.
Yes.
Not 6 years.
Biodiversity. Yes. No, but I have heard about
Dr. Khan.
Secretary Nematology.
The most active man.
Most active man.
Pure,
Where is he based?
pure.
Where are you based? Me too. কেমন আছেন? হ্যাঁ,
Okay, so my voice is good. We're going to start like 5 minutes time. We are waiting for Dr. Gaur to come. All right. I don't think I need a mic, right?
ओके फाइन। नहीं ठीक है बहुत अच्छी बात है। आप लोग सब कर रहे हैं काम, तो अच्छा है, बहुत अच्छा है। और हमारा तो मैथमेटिक्स स्टैटिस्टिक्स में जो है अगर मिल सकता है पूरा बन सकता है, बहुत अच्छा है। No, but that's why these days, but that's why these days collaboration is becoming important. Those amounts of data, something, मतलब tangible अगर outcome it can be designed, that would be very good. Please, I respect.
अभी आप यही
স্যার দেখে নিন হচ্ছে ইউটিউবে চলে গেল
Those who are not able to come, they can see online.
So this program is being live casted in YouTube and in Zoom for the inaugural part. And at least one session when Dr. Sharma will be talking from Australia. After that, we will be offline.
Yeah, Patel is coming, sir.
Patel sir will be online.
Online.
Right. Up to it.
Yeah. The recipient.
President of
President of NSA.
faculties of the division.
Faculty of the Division of Nematology. Where? Yeah.
The IRA nematology is basically the kind of
is the core group.
we have also the project coordinator.
Oh,
right.
Yeah, we
So we have like a group, AICLP nematosis. For meeting that we did, when we talked about it, that is the same group. So now he is heading that.
So this, I was just telling him also that if some tangible outcomes can be thought of, and the things can be found with what towards that, where ISI can come from.
In my talk, I'm not talking about AI and touch from my
AI
side.
and it can be formalized rather than giving it at a formal level where things don't matter.
We have a dedicated group here. I'll show in my talk. We have done, scratching the surface and nematodes.
Find out very right. So it's whatever steps we can take.
Let me know. I'll invite you. We can have a.
I know in case of nematodes it's a bit difficult than
We are actually starting. No, actually we have started and it's working nice. We have started with like Carnicola and Computer and Behavior.
Yeah, but
And it
the
has actually
domain
able to
specialists, they have
identify
to
or
actually define
somehow
the problem.
nicely. But
We,
how
data
it's
scientists have no
identifying
idea
is the
what
big
it is.
question.
Yeah.
That is sort of a black box. Yeah, I know. That is where the research is needed. I cannot pattern from that. Maybe we will discuss a lot. Glad you can.
He enjoys AI.
Oh, of course. Yeah.
National level policy.
Great to see you. For a long time.
नमस्कार नमस्कार कैसे हैं आप? This is indeed we are proud of this institute, and that's why we want to see if we can. Is it? We are not exactly a deemed university, and we call it Institute National. At the time, like 59, we were 59. And I'd say,
Many things happen.
Many institutes,
So,
some
we
started.
Research
Research was taken out of the university.
So good morning all. I hope I'm audible well to you. And it's a pleasure to host once again a August forum of nematode researchers in ISI. We did it I think 7 to 8 years back. This was a small group, but that was the beginning of my interest also. And one of the greatest pleasure I'm having standing here is because I'm working with one of my senior teachers who have taught me nematology when I was in MSc. And now working with him is giving me, it's a feeling I cannot really express, honestly, with you. So once again, thank you very much for all of you to showing interest in the National Symposium on nematodes done for Sinfos and Friends of Plants and Soil Health. So we're going to start the inaugural program. I would like to call up on stage the Secretary of the Neurological Society of India, Professor M.R. Khan. Professor Khan was my teacher in master's degree, and he still is a teacher. Now I want to call up on stage Professor Shirohi. Professor Shirohi is the President of the Neurological Society of India. Now I have the pleasure to call up on stage the special guest of this inaugural program, my director, Professor Shambhavita Bandyopadhyay. Professor Shambhavita Bandyopadhyay is an esteemed computer scientist, very well renowned in her field of research. She has earned many accolades in her research domain. Mainly, she is the recipient of Shantishore Bhatnagar Award, TWS Awards, Infosys Prize, and recently she has been conferred with the Padma Shri Award for her contribution of science. Now I want to request the chief guest of this program, Professor Shapon Datta, to please come up on stage. Professor Shapon Datta is a distinguished plant scientist, brings extensive expertise in plant biotechnology, agricultural science, and global agricultural policy, including IPR. Formerly, he was the Vice Chancellor of the Vishwabharati University, and he was also the DDG ICAR. He has held key roles globally, from being a senior scientist at Zurich, ETH Zurich, Switzerland, to serving as the Harvest Plus Global Rice Crop Leader at the International Rice Research Institute, Philippines. Recognized as one of the top 25 Indian scientists in his research domain. Now we have once again the distinct pleasure of inviting the guest of honor, Professor H.S. Gaur, in our dais for the inaugural session. This is the second time I'm having the pleasure of. This is not a guest, I think he's correct. Professor Gaur, as you all probably know, is a distinguished nematologist, researcher, educator, and academic leader, currently serving as the distinguished professor in the School of Agriculture, Galgosia University. He has an illustrious career spanning over 47 years. He has held key positions including the head of the Division of Nematology, Dean Postgraduate Schools, Joint Director ISAP, and many things. So, I would request the President of NSI to please felicitate our Director, Professor Shambhavita Bandyopadhyay.
Thank you very much. Start like this, and we look forward to such interactions. Fantastic, very nice. I think it is from some places. Thank you. Thank you very much.
I once again request Dr. Shirohi to felicitate Professor Shapon Datta. So, Professor, I request Dr. Shirohi once again to please felicitate our Dr. H.S. Gaur, the guest of honor. request Professor Khan to please felicitate Dr. Shiri. I request Professor Khan, the Secretary NSI, to deliver the welcome letters.
Good morning to all of you on this auspicious occasion. I have the great pleasure to welcome you on behalf of the Nematological Society of India and my own behalf. It's a great moment and my privilege to welcome these great people from this country. I can see that some of the people who are our teacher, illustrious teacher, and also some of the people who are distinguished in their own field. So I first say that the dignitaries on the dais and the representatives from the government department, the senior scientists and these students present in this conference to welcome you all. And also it is my privilege to have the gratitude to all the dignitaries present on the dais. We are thankful to these all renowned people like here, Professor Swapan Datta. Sir was very much kind to accept my invitation over the phone and he immediately agreed to come on this occasion. I did not to introduce him because already Dr. Abhishek Mukherjee has already told about him. Today with us we have Professor Sanghamitra Bandyopadhyay, Director, ISI Kolkata, our own teacher, Dr. H.S. Gaur. Sir is holding the key position in nematology in IRI as a joint director as well as dean. Also, he holds the vice chancellor of Sardar Vallabhbhai Patel University, and currently he is also very active. Sometimes my teacher used to say I am younger than him. So today his presence is a great source of inspiration for us that these three generations of the nematologists are here, and Sir has come here and joined us in this program. It is my proud privilege to address this gathering by saying few words about the Nematological Society of India. This society was established in 1969. Now we are going to complete almost 55 years. So society is promoting the nematology science, education and extension in the country. Also, the society have this flagship journal, Indian Journal of Nematology, started in 1971 and it is publishing continuously without any break. So this is our society's commitment to the science and this promotion of the nematology in the country and outside India. Society also have this great role in disseminating the science and giving the platform for the all generations of the nematologists in the country by having the discussions, presentations, so that we can bring the science to the forward. So we need to update ourselves. Therefore, this society is organizing several symposium. So far, 24 national symposia, conferences has been organized in the different parts of the country. And this year, it is our, say, proud moment to say that this year we are organizing the national symposium on nematodes, the unseen foes and friends of plant and soil health, for 3 days program at ISI. So this is a great institution, and we never thought of having this kind of program in such a national institute. And this platform is to giving this platform to share the knowledge and exchange ideas, foster collaborations in the nematology science. And this is also giving some of the latest advancement in the science of the nematology and giving some of the management practices and sustainable agricultural practices through different deliberations. I would like to happy and to inform you that we received overwhelming response in this symposium. We received 76 posters, 20 invited lectures, and 2 oral presentations on D'Souza Memorial Lectures is going to be presented by an eminent scientist, Indian scientist who has been recognized by the society. And also there are several award presentations where the young scientists are going to compete for the most prestigious award of the society, Professor D.J. Rasky Award and Dr. Sudarshan Ganguly Award presentations. Sir, we are happy that this symposium is being represented by the scientists from the different universities, institutions, and we have the 11 technical sessions. Some of the prominent sessions of this symposium are the advances in nematode identifications. We have the speakers going to deliver the artificial intelligence and machine learning for identification, integrative approaches by using this molecular as well as the traditional approaches. We have the session on molecular nematological aspects by giving some of the advancement in this latest technology in the plant nematode interactions and also genome editing approaches for development of the nematode resistance in the plant. This symposium is also going to give a dedicated sessions on indicators on the soil and plant health, and also going to give some practical aspects of nematode management, and including the new molecules for management of the nematode. And this symposium is also going to dedicate on session industry and scientist interface. So this session is going to discuss about the new molecules those are going to come into the market, as well as some of the new formulations are only in the pipeline. So that will give some of the new advancement in the new molecule development, particularly for management of the nematode. Overall, this 3-day symposium will adequately address this latest development in the science, and also we are going to plan for the future nematological research to make our prepare for the future for the food security of the country. I thank each one of you for your kind presence, and I look forward to making this event successful. Thank you, sir. So next we have this lighting of the lamp as per this customary of this kind of program. So I would request to our dignitaries on the dais to light the lamp in front of Professor Mahalanobis, PC Mahalanobis.
We'll all come together, but we must. पहले आप करके गाड़ी निकल जाएगी. I've already did it.
May I now request Professor Shireen to deliver that.
Professor Supun Kumar Dattasi, Madam Bandyopadhyay, Professor Gaur, my colleague Ahmad Khan, Project Coordinator Dr. Gautam Chawla, heads of different nematology sections of Indian nematology departments, Professor Walia, Professor Jain, senior colleagues, other scientific colleagues, and I think we have a good large number of students. I think I feel privileged and honored to be on this dais where we have one of the top academicians of this country. I think as introduction was given regarding Professor Supun Kumar Datta, he's been really vital in deciding future of research in several areas at ICAR and the universities which he has headed as vice chancellors. His international experience and his special findings and transgenic research in rice, I think, is marvelous. He is the man behind the golden rice, which is enriched with vitamin A. Then I think ferritin also, sir, iron-enriched rice, BT, you name it, and he has a long list of things. And the kind of experience he has had in international arena is tremendous. And ICR as well as the other universities have really gained with his experience. And we are really happy to have you here, sir. And thank you so much for agreeing to be here with us. And I think your presence will definitely give us new directions to work in nematology. And ma'am, I was reading about you a bit, and the government of India has honored you with Padma Shri. I think that speaks all the kind of achievements you have all across. She also has lots of international experience. A lady with computational biology knowledge as well as bioinformatics, and I think that's where we are looking forward for some bridges to be made with this institute as well as our nematology fraternity, be it IRI or be it other departments and all that. And Professor Gaur, my teacher, river teacher, a man who sleeps and wakes with nematology, ma'am. He is one of the most passionate nematologists we have in the country, and he has been successfully running nematology. We all look forward for his advice and consultations whenever we are in need of. And he has established nematology in 2 universities. Earlier, he established at Meerut also, where he was the vice chancellor. And at IRI, I think we've been seeing for all the time. Thank you so much for being here with us and really inspiring all the young as well as the old colleagues. And we happen to be colleagues, so I don't think Ahmad Khan needs any introduction, but all his efforts and Professor Abhishek's efforts have led to this function in shape today. And I think the deliberations would be really wonderful. That's where we look forward for. And though I have a small little time to speak about, but then some of my observations with respect to nematology, I would like to place on record. Because we have had a fantastic launch of nematology in this country. If you look at the last 30-50 years, I think the previous generation really gave us a beautiful platform where nematology departments were established and the research was there. And we are reaping the fruits of that base today. But today a bit things are different. But anyway, if you look at it, we have had some fantastic research outputs with respect to management of nematodes, be it in the biological control area, be it in the molecular biology area, be in the ecological area of research. So those are things which we have already achieved. But we do look forward that today, if we look at the requirement of the day, we need to increase the food production by at least 2% annually if we have to keep pace with the demand of food the way our population in this globe is rising. And today, if the data is to be believed, around 30% of the loss to all food crops happens because of biotic stresses, nematodes being one. Though we may be undervalued, but I think we do have a lot of nuisance value in damaging the produce of food crops. And that's the reason that we need to look forward for future research today.
in areas where we should think of, because many things have come. We cannot add on the agriculture area or the area under cultivation, but then yes, we can evolve research and cater to the newer challenges which have come because of the global climate changes. So we need to look at research in different perspective today, considering that the climate will change, soil resources are going to deplete, water is going to deplete. All these parameters also need to be taken into account if we have to go for a research tomorrow. Biological control is one of the preferred modes where, because it doesn't get you that kind of a toxicity which comes with nematicides or chemicals, but then here also we need to understand the interaction level. Because if you look at the parasitism of nematodes, unless you really understand the talk which is going on between these two different kingdoms, one is the plant and other is the animal, only then we can come out with solutions which can give us a fantastic or a tool to manage them. And that's where we need to understand even the biological interactions with the antagonists of these nematodes and the crops at molecular level. That's where I think we should look forward for. Moreover, when we look at the research, we usually try to understand and compare only one parameter of it. But today it's a complex thing. Soil is a very complex media which is being governed by lots of biodiversity organisms living inside. So we have to expand and we have to look forward for research where all these sort of considerations are taken into account. How populations are changing of the, or rather microbiome is changing. And let me tell you, microbiome is basically most impacted by the plant species or the crop species which you are cultivating. So even that can be managed and organized and planned out how we can improve the antagonistic population of the microbiome which favors or allows nematodes or suppresses the nematodes. So that's again an important area which I think all of you or students can plan about. Genetic solutions, though we have been able to identify and clone several genes of resistance, though may be limited, but yes, and some of their transformations have been successful. But the problem is the policies. Because of policies, we are unable to release out anything in the field for the farmers. So that still is a concern. Say, giving an example, BT brinjal is being cultivated next door in Bangladesh, but it is not getting cultivated in India. So policy matters are a big issue today concerning that. RNAi has been able to give us management strategies by delivering psRNA to the host and then possibly which is getting picked up by the nematodes and we can attack and hit the crucial parameters of nematodes. But that again, though there is no synthesis of proteins, but again policy issues are there. Several RNA based GM crops have been released outside, but then again we are still holding on. Or rather, our lab has patents on promoters which only allow expression of the genes in the roots on infection by nematodes. So those are really important things which can definitely change the future course of nematode management. But luckily today we have options of using genetic engineering, using the CRISPR-Cas tool for genetic editing, and possibly that will give us way out at molecular basis to manage nematodes. Then areas like ecology are also important where we need to study the impact of environment, climate onto the nematodes, and that's again in a complex manner. So we need to look forward for that as well. Government of India is projecting lots of drones technology for farmers, and drones can also be used for capturing the images, thermal images, wherein you can try to identify whether which nematode is a problem out there. Already researches have been made where rotalinculus fields could be identified by going for spectral analysis. I think that's one area where we can also take help of this grand institute and two of people of kinds, a nematologist and the data scientist out here can help us evolve that sort of strategies. Because evidences are there that spectral analysis can be used for nematode disease identifications. Then definitely studying the interactions, both biotic as well as abiotic is important. That's what I said. So I look forward that you will take these challenges for tomorrow. But then again, there is a small little chapter which I feel that over a period of time today in many universities, the strength of nematologists is depleting. And that is resulting in people taking less interest into it. Somehow we need to recover from that as well. We need to show around the importance of nematodes. We need to have more demonstrations at farmer fields. We need to have a stronger extension so that we understand. Plus, I believe we need to have a system of diagnostics at state levels, at farmer levels, at block levels, where possibly the farmer knows that my crop is going to get infested because of these nematodes, which are though invisible but really damaging, having a damaging potential of very high. And I will thank Dr. Walia taking up steps for the Ministry of Haryana, Ministry of Agriculture of Haryana state, where it is being made mandatory. If somebody wants to take a subsidy on your polyhouse and all that, greenhouse cultivations, they will have to first get the soils tested for nematode populations. Because ma'am, the spread of nematodes is basically through nursery or through soil and through roots. So that's where, if we have that sort of system, that the nurseries of this country get licensed or certified that whatever material they are selling to the farmer is nematode free, I think the major spread can be checked. And that should be there with respect to other pathogens also. It's like we look forward for some standards with our health that is there. So these are the areas which possibly we look forward to debate upon, look forward upon, and assess where we have achieved. So with this, I thank everybody on this stage as well as off this stage for being here, and with your support, we will be able to make this symposium a grand success. Thank you.
Thank you, sir. May I now request Professor Shanmugita Bandyopadhyay to deliver an address.
A very good morning to all of you, and it's a pleasure to be here in the inaugural session of the National Symposium on Nematodes, the Unseen Foes and Friends of Plant and Soil Health. And it's my pleasant duty also to welcome all of you to the Indian Statistical Institute, which is hosting this symposium. In particular, I thank Abhishek Mukherjee and all colleagues of the Agricultural and Ecological Research Unit who have played their very important roles in organizing this activity here. We look forward to such activities because Indian Statistical Institute was established with the very basic philosophy of the principle that statistics and mathematics, these are the basic tools which have to be used for various other natural and social sciences. That is the objective of this institute, that statistics as a tool has to be used for the benefit of various natural and social sciences, and those sciences also should challenge statistics. Now, in today's world, with statistics and mathematics, we cannot but add computer science. Computation has to be there, it is there, omnipresent. So our objective of this institute is slightly now broadened, that dissemination of knowledge of statistics, mathematics, as well as computer science, and their applications in various domains, including this very important domain because it directly affects all our lives, the soil health. Soil health is extremely important for each individual, each one of us. So we look forward to a very interesting interaction of exchange of ideas over these 3 days. How data science, how statistics, mathematics, and computer science can help analyze the data for you. But the problem has to come from the domain specialists. You have to define the problems for us, and sometimes it also happens that once the problem is defined, before data collection, the experts can tell you exactly how to collect the data, what data is required to be able to answer that question that you have. And therefore, this interaction should not start after data has been collected. It should start before the data has been collected, at the time when a problem is being visualized, is being seen. Some questions are being formulated. From that very time, this interaction should start. It is easier said than done, because in India in particular, we have struggled to collaborate. Even within an institute, within a department, we struggle to collaborate because we take pleasure in working in silos. Maybe we are very worried of who will have the authorship, who will have what position, who will take the credit, etc. We are so much worried about that that we forget that we have to solve problems that affect us directly. So that is where I hope our minds will open up and we will start interacting in much larger doses. ISI, as you know, you've seen the picture over there, ISI established in 1931 by Prashanta Chandra Mahalanobis, who was himself a physicist by training. He taught physics till he superannuated in Presidency College. But while his studies of physics was going on, it was a chance interaction with statistics that led him to believe that this particular discipline will be essential for the development of the nation, for human welfare. And that is how, with some common-minded people, he started the Indian Statistical Institute in Presidency College with a paltry sum of 250 rupees budget, on a budget of 250 rupees, which now has grown more than 10 to the power 7 times. So, and also started in a small laboratory in Presidency College in central Calcutta. It moved to its present campus in the 1950s. The headquarters are here, but we have centers in Delhi, Bangalore, Chennai, Tezpur, and we have a branch in Giridih where primarily agriculture research goes on. We have campuses of the Statistical Quality Control and Operations Research in Hyderabad and very small presence in Mumbai and Pune. So we have more or less a pan-India presence. We have, of course, our constraints, but this institute, which we are proud of the legacy of this institute and also the currently whatever is going on. If you really look or dig deeper into the awards which come to this institute every year, it's actually, given our size, given our limited faculty strength, it is very large. And we struggle to, first of all, of course, we struggle to live up to the legacy which has been left behind by our predecessors. Professor Mahalanobis, C.R. Rao, J.K. Ghosh, many such people. And, but we also try to make legacy of that stature so that our successors will struggle to live up to this legacy. So it should not only be in the past, but we are trying to make our present also as strong as possible. And with all the colleagues everywhere, pan-India, we are trying our level best. We really hope that the Nematode Society of India launches, or the nematologists in this country, they launch some national level programs where people of different expertise can come together, and I'm sure ISI, especially led by Abhishek, who's in-house, and other statisticians, data scientists, computer scientists, mathematicians, there will be a few who will come forward and share the load of solving such national missions. I am not sure if there is a nematology or a nematode map of India. These, if it is there, it is fine, but these sorts of things are very important for any data science program to succeed. It need not have any question immediately, but just to map it, where, what sort of population is present, etc. Just such mappings will be essential. And I'm also not sure if such data is available, if it is there, whether such data is available freely for use by any researcher, any student who is interested. Because only then can, somewhere in some college, maybe can formulate very interesting questions. So making data available is also a very important challenge, where also in India we have struggled. We work with data of other countries, but for soil data it will not work. It has to be Indian data. But in many other domains, what we do, I work in computational biology, We constantly work on US data, European data, Chinese data, we lack Indian data. It's not there, but the realization is there. And now things are sort of different organizations are trying to change the state of affairs. And artificial intelligence, machine learning, we are right now seeing the revolution in AI in front of our eyes. It's a revolution unfolding right now with the large language models over just the past 2-3 years. Remarkable changes have happened, and that is going to change our future in ways we cannot imagine today. But AI, machine learning as a tool, especially generative AI, where you can generate from nothing. You can generate text, you can generate images, you can generate speeches. For large democracy like India, it's also a real challenge, it's also a real scare. And because regulations are very weak. And as for nuclear energy, even for this particular tool, the artificial intelligence machine learning, it will depend on the hands that use it, on the minds that use it. It can be put to great use, it can be put to great harm. So how we use it is very important, but the march of research in AI and machine learning, good, bad, whatever, cannot be stopped. It will continue, and therefore, with regulations in place, we hope we can also, because Indian Statistical Institute has core strength in AI and ML, and we hope we can talk to you more in detail during these sessions and also thereafter. Very important that those seeds are sown in such interactive sessions, which I hope will happen over the 3 days. And with those few words, I again extend my heartfelt welcome to all of you to the Indian Statistical Institute, and I hope you have very enriching 3 days of deliberations, and with your questions, you not only will you be enriched, but we hope as an institution, Indian Statistical Institute will also be enriched. And I was just talking to our chief guest of today's function, Professor Datto, that my experience has shown that things really start working when we take it towards some intellectual property protection into some sort of business model. I am not sure if that has happened in the nematology domain, but it's high time that things are patented, things are put to use, maybe in a business model. Those young students who can, please keep this consideration in your mind, that while fundamental research, fundamental science has to progress, we have to do research there, but those who can, please also consider entrepreneurship as another career option, not the only, not everybody can do it, because it's not easy. I can assure you it's not easy at all. But those who have that mindset, who can, please take up, please think of entrepreneurship in this domain also, as your career plans, because if we, in 2047, if we really want to become a developed country, then we have to generate knowledge and hold knowledge and convert knowledge to wealth within the country. Thank you very much and wish you all the very best.
Thank you very much, Madam, for the inspiring words. May I now request our guest of honor, Professor H.S. God, to deliver his address.
Good morning. Esteemed Chief Guest this morning, Professor Sapandata, the Director of this great institution, Madam Shri Dr. Sanumitra, Dr. Shroy, the President of the Society, General Secretary Dr. Imran Khan, a very senior colleague sitting in the audience here, Dr. Bajaj, Dr. Walika, Dr. Lengaras, Dr. Hong Kong, Dr. Kursia, Dr. Chaube, and many others, and all very dear colleagues of mine and my students who are sitting here today. It's a great occasion, I feel, to be present. We are also present at an institution which is very closely connected to us. When you say nematology, statistics is not far behind. We began with numbers. In biological system, when we started understanding populations, understanding distributions, the population growth, models, crop damage relationships, the fluctuations in populations, even demography. Our models depended upon the nematode numbers. If you read some of the old books on statistics, Snedeker and others, Cochran and Cox, you will find data from nematology used in the development of those models and their illustrations. I particularly began my career by using statistical models in nematology way back in 1970s. We had the chief guest, none other than Professor Datta, is a biotechnologist, and biotechnologists have a great relationship in nematology. We know that CA ligands has been used as a tool in biotechnology and genetics, being the first organism to multicellular organism to be genome sequenced completely. And a lot of developments have taken place after that. The particular kind of development, the ontogeny systems, the function of every cell or every gene, that could be understood, and the fate of every cell in the embryology. That is where a lot of connection comes here. Friends, nematodes themselves are typical organisms who are the middle stage of the experimentation by God. We have the protozoans, and then moving on to red helminths, and then helminths, the nematodes, the pseudocoelomates, and then the coelomate animals. Pericoelomate, pseudocoelomate, and coelomates. And how the organ functions, how the metabolic functions, they change over the evolution. That is the subject that we deal with. This is a very, very important occasion to have this symposium, a very important topic when we consider both nematodes as both friends and foes. We have been mainly focusing on nematodes as our foes for many, many centuries, many decades. But the realization has also come that they are not only foes, many of them are our friends also. They need to be conserved as well. So we can use nematodes in various dimensions. This particular symposium is also very important for one more reason I am finding here. We have the head of the division of nematology, the project coordinator of nematology, and the president of NSI together on the same dais, and the great professor Dr. Amar Khan, and many others. This is a team which has to lead nematology actually at the national level. And when they come together, nematology has a great future, I'm sure. Nematology as such, has grown in various dimensions, sometimes decreased in some areas, but increased in many others. Actually. We began with simple descriptions of nematodes. We began with morphology, we began with scanning electron microscopy, went to statistics and biochemistry and biotechnology and genes and these functions, RNA and other things we have been discussing, barcoding, metabarcoding, other things going on. And computer applications, particularly the use of AI, will be a great necessity in nematology when we understand the complex variations in nematodes and how to come out with a real clear decision on what particular species is, what particular population is, and how it's going to behave. Very minute differences that we can, but we have to take this as a action only after knowing the correct identification or correct decision making. Because there is a lot of variability within species also. But coming over that species variability in many factors, there are some parameters in which they become distinct. And that's where their behavior changes. And here, the use of computation and AI is going to be a great tool in the future actually. Nematology, we always talk, nematodes, I used to say nematodes are a problem of bad agronomy. not understanding their environment, not understanding what changes their populations and behaviors. Many technologies are developed and where they can have counter effect on nematodes, particularly as pests. People are talking of zero tillage and everything. I have been always opposed to that. But it's necessary. Zero tillage is fine, conservation agriculture is fine. But we have to find ways to tackle nematodes also side by side. How we can merge those things together, and what are the changes in ecological parameters that we can make so that they don't become a problem. At the same time, we can have good produce and save our environment and also economics. There will be many things to be talked in the technical sessions in this symposium. I don't want to take more time here, but you and the chief guest will be having great words from him. I know Professor Sapundatta from many years now, and I have not seen a more humble person than him and the most knowledgeable person than him. Any institution where he has gone has benefited from his presence. ICR was a great beneficiary. We ourselves were great beneficiaries of time. And now the two universities, Vishbharati and the other one, which is the founder of Essential, Vishbanga Vishwavidyalaya. So this is, they are the great institutions to be benefited by them. But indirectly, many of us still continue to benefit sir, from you. Thank you very much. Thanks indeed. Thank you.
Thank you, sir. Thank you very much. Before we hear from our chief guest, Professor Shapun Datta, may I request our guest on the dais to please release the abstract booklet formally. Which is being printed. So as you know, the abstract is already been released as a PDF document in our web page, and it's accessible from there. If you can print, if you want to print, you can print it from there. So now, I would like to request our chief guest, Professor Shapun Datta, to deliver his address.
Good morning to everyone. We are very pleased and glad that we are here today to discuss the unseen foes and friends of plants and soil health, particularly in the context of nematodes. We are here at Indian Statistical Institution. It's a great institution, founder D.C. Mahalanavish, as we heard from Professor Bandyopadhyay, the director of ISI. We are with us today Professor Shangamitra Bandyopadhyay and Professor Anil Shirohi, the president of the Nematological Society of India, Professor Matiar Rahman Khan, the secretary and the very dynamic person on nematology research and administration, and Professor H.S. Gaur, many years we are together at ICAR. I'd like to see Professor Gaur, a nematologist in profession and research, with us. Dr. Abhishek Mukherjee, the local secretary. And I see many distinguished nematologists, scientists of India. They are here, research scholars, students, and many other those are participating today and tomorrow. The most heartiest welcome from my side that you all together here today and next 2 days to discuss on nematology and its future. I'm wondering when I accepted when Professor Khan asked me to come over here and to give a talk. But after that I started thinking that it's a meeting of nematology and entire things on nematology, and so why I have been chosen to be chief guest for this particular meeting. I did have little work on nematology when I was working at International Rice Research Institute. I'll tell a minute on that. But that's not the reason that I should be here. But then I probably thought that I knew Professor Khan for many years, and I have once interaction with him on nematodes in an interview. Or maybe I'm one of the oldest scientists now, and so that he thought that I might be invited to be there with you. So nevertheless, it was my pleasure. While I was at ICR, I didn't look that much on nematodes, honestly speaking. But last few days, I really enjoyed reading nematology and literature. And Dr. Khan gave me a huge amount of literature. I asked him, and he provided me very generously, even unpublished book also he sent me, and I had time. And I took interest because I knew when I was student of biology, C. elegans, and modeled material for the biological studies, better part the molecular biology, but did not know much exactly what it does and what it about. So thank you, Dr. Khan, actually, that you allowed me or you gave me the opportunity to study on the subject. So what I felt as standing here today with you, the nematodes are simple biological material with few cells, but with that few cells it does all the vital activities. Span of life is very short, so all the biological experiments and everything can be done. And particularly with the today's, with the C. elegans, with the only 302 neurons and about 7,000 connections that govern the whole body, meaning that today is the nervous system, all the brain work, and even a live Lego has also been built up based on this model material. So anything now, everything, what you are looking for the new dimension of science, starting with the C. elegans or other nematodes, that today it is possible. And with the, you have already heard or you have been looking at the artificial intelligence, and the potential of future use is enormous. Anything or everything can be used this model or Lego as a model for future studies. Now, why we did not pay much attention, and today we're giving definitely more attention on the nematodes. I think as nematodes, we knew that from the even Veda's time, 9,000 years back, as a creamy worm. Even it is mentioned in Hippocrates, it's mentioned in the Aristotle, it is mentioned in the old Bible. Everywhere it is mentioned. So it was known, but we didn't pay much attention that what we have to do with this nematodes. It live in soil, plants, even desert, or even marine conditions, lithospheric. But as it is there, sometimes as you know that বায়ুর সমুদ্রে বাস করে আমরা বায়ুর অস্তিত্ব বুঝতে পারি না, যে বিখ্যাত একটা স্টেটমেন্ট আছে, living in the air, we don't understand what is the value of the air. So these nematodes are everywhere. It's causing damage, but we don't see this because mostly it remain in the root rot and under the soil. Now there are two things today is striking to me. The title of this symposium perfectly it shows the importance, the unseen foes and friends. So that beneficial nematodes, what we see in the soil, and today you know that Warren Buffett and Bill Gates, two very rich men in the world, they made a foundation, and that foundation, they have different things, but one of the foundation, they're working or spending billions of dollars for this brown revolution. That means they're entirely working on soil fertility. And now I see a very nice connection to the nematodes. The nematodes, basically in the soil, the debris, whatever available in the soil, they break down and mineralization resulting carbon and nitrogen. And that means it further recycle in the soil structure and soil fertility. I'm sure the project is still not in public domain, but they have been working for a few years in a very restricted way. I'm sure nematodes would play an important key role on that soil fertility and soil structure reformation. Now, as in this context, as Professor Shangamitra Bandyopadhyay was mentioning about the AI and Indian Statistical Institution. Now, this meeting held in ISI, and whether this ISI means we always consider that statistics, maths, modeling, mathematical modeling, etc., which is true. Although Prashant Mahalavish even Tagore even invited him to work at Vishwabharati based on his work on water. That's a different context. But anyway, that's not the issue today to discuss. But the ISI's involvement and artificial intelligence, these two together, with the parasites or the nematodes, parasites in the soil or in the plant, or enormous damage. At the same time, they have a possibility to make better soil or soil fertility. Here, ISI can play an important role because the population density of the friendly nematodes, the critical density or critical information is missing. I looked at the different literature, but always it is referred that they do a good job, they're beneficial, but how it does. I have seen many other recent and past research papers, so the different microbial population density is very much needed in the soil to maintain the soil ecosystem. That does lots of other things, including biological nitrogen fixation and etc. etc. But the nematodes population, how to use it, and that means it's very easy to maintain this population, and then so that this population, the friendly population, need to be incorporated into the soil. As today we are having a different agriculture practice. We are having, greenhouse or the protected cultivation, etc., etc., and in that way that the plantation and the agriculture practice is governed. It is very much well structured. Now, in this well structured condition where the high value vegetables and other crops are being grown, so where a population of friendly population of nematodes can easily be admixtured or can be incorporated. Now, this population, the statistics and everything all together, and the algorithm and the entire things need to be understood. What I say, we need to do a research. That research component is missing. So the new project need to be ingested, and these new projects need to be developed in the context of this population density and the statistics. The data, the real data need to be accumulated, real data need to be utilized. So I would urge the young generation or young scientists to come more into the nematode studies or the nematode research, or to do no matter whatever disciplines you have, you do a more collaborative research with other disciplines people, and then develop a project in a comprehensive manner, including AI and also the statistics and also the dynamic nature of the nematode application in the soil. That would be the most benefiting to have this meeting at ISI. And I find this would be an interesting integration that ISI and the Nematode Society are coming together and to think about in the next 3 days. One of the things that, as my subject is biotechnology, so I definitely would say a few words on that, particularly that it is well known that the cryproteins has a tremendous impact on lepidopteran and other insect pest protection. But the cryproteins are larger in size that cannot feed it or pass through the feeding tubes of the nematodes. So what do? here comes the protein engineering. The protein engineering can reduce the size of these proteins, and these proteins and some other peptide, other chemicals can easily be incorporated into the nematodes to destroy that particular food pathway as well as these particular structures. Otherwise, the proteolyze or the trypsin inhibitor proteins and many of those genes, lectin genes particularly, snowdrop lectins and other lectins which are known that biotechnology people are using it for protecting plants against the fungi. And as one of the things I must tell in this context that plant and fungi or these parasites, they come together, they grow together, they live together. You cannot control them 100%. Better you don't even try to kill them 100%. They would never. More 100% you want to kill them, there would be more resistance sort of things would be developing, more different strains would be developed, and that would make even havoc. So we know that they live together, they would live together, but the management strategy or the scientific intervention would be used in such a manner, we delay the symptoms, we control the symptoms, but it not affect the yield. It would not affect the ultimate, the production what we want. At the same time, the beneficial aspect also we can trigger more to happen in the plant, so the plant growth will be bigger. More bigger plant growth comes, the parasite or nematodes or any other pathogenic parasites would be less and less. So it's a management, the integration of the management, the proper management, scientific management, and at the same time biotechnological intervention, and possibly if you can develop resistance host plants. Resistant host plants by genetic engineering or by RNAi technology or by genome editing, CRISPR-Cas and other things what are available today. All of these things together can be integrated. But I think the people who will be working on nematodes or its future direction, they got to be multidisciplinary subject knowledgeable people, so that they can understand soil, they can understand plant growth, they can understand agronomy aspect of the soil where the plants are growing, at the same time the new tools of whatever available today. I think today's science particularly, you can be specialized in one domain, but you have to expand your domain with understanding of other discipline subjects so that you can integrate it in a better way. So this is the possibility that I find this symposium could play an important role. If you look at the economics and the new chemicals or the molecules are coming in the field, that also enormous work is going on. I learned that tomorrow there will be interaction with the private sector, so where different private industry people will be making their presentation. There are a few chemicals that are today very important for protecting the nematodes as nematicides. But to me, it would be extremely difficult to control nematodes unless, with that chemicals, of course, that would help, but other management strategy also need to be incorporated together. I think as Indian agriculture is moving in a very dynamic way, today we are in a position that exporting more than $50 billion agricultural export, which is enormous, and it's expanding. Now, as our agriculture cropping intensity is more than 130%, it's also increasing. I think we can't go beyond that, and that's where we reach to a certain level that we have to have a better understanding of the management. Now, with that, the agriculture economics that we see today with India, even feeding the Indian population, still we're exporting rice, wheat, and other crops, and it will be more and more in that direction. And with that also, with that demand of more crops, more food, more vegetables and other fruits to be produced, more parasites, more nematodes, more pathogenic fungi also would be coming into this condition. And that's the reason we need to protect the plants against those pathogenic fungi as well as nematodes or other pests. And for that, I would say, as this is a good thing, there is a demand. It's a good thing the private sector is also coming into the picture. As Professor Bandhavadhyay also mentioned that there should be a trade-off, there should be some trade interest incentive is required in doing science and utilizing science and applying the science. So I think the platform is excellent. With all these things together we have in place, and the future direction should be that integration of these different discipline and science, including today's AI. And also I need to make one cautionary note also. The AI, the way AI is coming, it's generating lots of information. Many of the information are not right. So this information also need to be carefully judged, that whether we are getting the right information to follow. Rather keeping everything in perspective, we need to judge and need to utilize it, the right database as well as the AI, and integrate the science and management together to control the nematodes, to control in the sense also beneficial as well as protection against the pathogenic type. I think that would be the right sort of way of looking the next 3 days of this symposium. And once again, I welcome all of the participants for this meeting. And I'm sure that you would interact and you would play vital role, or please do ask questions and concerns that you may have, and to make it interactive and more dynamic session that in coming few 3 days. Once again, thank you very much for inviting me to give this opportunity to interact with you. Thank you very much.
Thank you very much, sir, for the encouraging words. It's my duty now to deliver the vote of thanks and close this inaugural program finally. Formally, but before I do so, I just want to share a little bit of anecdote with you. We are discussing a lot of applications of AI and in nematology. So last night I was preparing my slides for today's presentation. Right, it was the I had to do it last night, and while doing so, there is a app called ChatGPT. Many of you are probably aware. So I was just curious, and I asked ChatGPT, "Do you know any good papers that recently been published on AI application in nematodes? Can you give me those citations?" And ChatGPT immediately generated like 10 of those citations, perfectly nice, and all these beautiful journals and everything. And I was like shocked. How come all these papers are there and I haven't even seen those? And I start searching them, obviously with a lot of curious interest, and none of them Google search, Google Scholar search revealed nothing. And I thought, first thing I thought, maybe I'm missing something. I'm not searching it well. And go to the journal, go to the whatever they have given. There's nothing. So it's completely generated out of nowhere. Those papers doesn't exist. So as all of them are stressed while talking, AI has exceptionally power in interpreting things, but it has also a capability that we all need to be aware that it generates things that not really there. So you ask AI to write something about Professor Shangamitra Bandhavadhyay, it will write like 2 paragraphs. But if I give it to Professor Bandhavadhyay, she will like jump up. What is this? So anyway, first of all, in the vote of thanks, thank you very much, Professor Shiroi, Professor Khan, for allowing us to host this August gathering in ISI. It's a pleasure. ISI is one of those institutes, I think probably very few in India, where we have our agricultural side, albeit a bit weak, and a very, very strong statistics and mathematics and computer science side, working side by side. You don't see this often in other institutes in India. So the crosstalk is there, but it's very feeble, but we are increasing the crosstalk between statisticians and mathematicians. So this sort of conference helps us a lot to pass the message. I know Inaugural is here. Inaugural is a statistician, mathematician, is interested in this subject and want to know what's going on. So you see, that's why people are, people generate interest. The statistician generates interest in our field. I could not have, organized this program with help from my colleagues, my friends, my students. So many happy faces you see around. I am boasting now. Is all of them is our student from Giridih. Professor Datta was asking me in the morning, what is happening in Giridih? I was there was nothing. And it's a pleasure that we are doing some work. My colleague, Dr. Bhattacharya is there, he's a very accomplished soil scientist. I have another social scientist, Dr. Behra. So together we are working on different dimensions. We are developing Giridih, lots of people are getting PhD, and all of them are now here. They're inspired to be with you all. ISI staffs are really amazing people. I stay in Giridih, as you understand, majority of my talk happens over phone, that you need to do that, I need to do that, and everything falls in place. In particular, I want to mention about two person, I don't know whether he is here. Shunbir Chatterjee is one of the person he calls me probably in the night 2 a.m. also. You don't hesitate, because things to be done and things to be done there and there. So you don't care what's the time point. And because of this person, and there's another one, Shushant. Shushant designed all these, beautiful banners, posters, and he works behind all these, programs that we are, going on. So without the help of this, we have the artificial people there sitting. Shubha was there, Neeraj is there from the CSS people there supporting the computer. So all these guys, we all come together and try to make things working. This family feeling is still there in ISI, it's still hanging on. I hope it will hang on for longer time. And I would end it by thank you all for coming here, and you are showing interest for this symposium and making it a grand success, I hope. Before I formally close, there is two announcements. After this, there will be high tea outside. You can have something to eat and coffee to enjoy. And there is one very important thing that we have to do is a group photograph. Because there is so many people and many of them are not yet registered. So my plan is to do it at the lunch where everybody will be there and the place is a little bigger. So we will gather around, I will shout, you hear me then, and we'll gather around and get it done somehow. So please stay safe and enjoy your stay in ISI. Thank you very much.
So, sir, we will move to the guest house. We have some. And the registration desk is now will be open, so you can get it done who are not done yet. Thank you. We will start at 10:55. So one more thing, we have to start at 11:55. There will be an online lecture, and we cannot delay. Here only, everything is here only.
YouTube live streaming, should I close? YouTube live streaming, should I close? Or
For the time, pause it.
No, it cannot be paused. Live streaming. YouTube. Either it should be closed or should be look like this.
So are also going on.
Yeah, it is already online.
Hey, Neeraj, you can computer it. Yeah, लैपटॉप लगेगा.
लगा.
ऑनलाइन में आएगा तो यहाँ से हो जाएगा.
हाँ, ये मतलब वहाँ से प्रेजेंटेशन देना होगा ना.
नहीं, तो ऑनलाइन ही देंगे.
ऑनलाइन देंगे लेकिन वहाँ से ज्वाइन करना पड़ेगा सबको जूम में. अभी तो वो प्रेजेंटेशन यहाँ पे डायरेक्ट गाड़ी है.
हाँ, गाड़ी है. पीछे अच्छे ढंग का गाड़ी चलेगा. गाड़ी मैंने तो बुला लिया. এগুলোকে সরিয়ে দিও। এই নীরজ ভাইয়া, नीरज भैया इधर लैपटॉप देना पड़ेगा। हाँ हाँ, तो ऑनलाइन में होगा तो ऑनलाइन का प्रेजेंटेशन तो कोई उधर लैपटॉप देना पड़ेगा। এই সুদর্শন দা, এটা তো ওকে আছে। আগের দিন প্রশান্ত দাদা একবার এগুলোকে সরিয়ে দিও। আগের দিন পুরো প্রোগ্রামটা চালিয়েছি। রাখি না একটা সময় দাও চলে নিয়ে এটা কোথায় ছিল?
হ্যাঁ, বিবাবাড়িতে নিয়েছিলাম তো। কপিটা খাই আগে, কপি লাইন পড়ে গেছে, পালিয়ে গেছে।
इंटरनेट कनेक्टेड है? हाँ, तो प्रोजेक्ट तक तो ओके।
প্রজেক্টটা কি এই অন করো প্রজেক্টটা।
प्रोजेक्टर चलो तो इंटरनेट इतना स्लो है।
প্রজেক্ট অন করে দিয়েছি।
আলোটাকে ছেড়ে দাও, আলোটাকে ছেড়ে দাও, যে আলোটা ওর গায়ে পড়েছে আলোটার বাইরে বের করে দাও। জল দিয়ে
টেবিলটা এই জায়গায় থাকলে প্রজেক্টর অসুবিধা হবে না তো? প্রজেক্টর হ্যাঁ,
office are not easy. না না না, ওইটা তো পাঠালেই হবে। আমি মেল পেয়ে গেলে আপনাকে আমি করবো। হ্যাঁ হ্যাঁ হ্যাঁ, কি লাগবে? আপনাদের বিল্ডিং এর বিল দিয়েছে না বায়োলজিক্যাল সায়েন্স ডিভিশন আমি আজকেই যাবো ঠিক আছে ওকে न्यूज है लगा हुआ है उससे आवाज कैप्चर होगा ऑडियंस को उससे कुछ पूछना होगा तो उससे तो हमें इसको बिल्कुल रखना है लेकिन आवाज आ रहा है बात कर
હતું ને એટલે ઈ કે
એને કે ન્યાથી
Good afternoon, ladies and gentlemen. Let's begin the proceedings, actually the proceedings of the technical sessions, we can call it so, which are going to follow from this particular session and later. I request the General Secretary, Dr. Kumar Khan, to educate the members of the hall about the institution of this Gerard C. D'Souza Memorial Lecture and the contributions of this gentleman per se. Thank you.
Respected the chair of the session, co-chairman, and the distinguished scientist in front of the dais. So today we are very happy to have with us Dr. H.R. Patel. He has been nominated by his Hematological Society of India as per the standard guidelines in the society to select a person who will be awarded the D'Souza Memorial. The body has considered his name, considering his
Dr. H.R. Patel, are we audible?
Dr. Patel, can you hear us?
Yes, now I am hearing, but it was breakable. Okay.
Would you mind sharing your slide, please? You please share
Okay.
your slide when you are ready, and we are ready for your presentation.
If the presentation slides, kindly share with us.
Yes, may I start now?
Yeah. Please do, please do.
Yeah. So, hello everybody. Good afternoon. Actually, I am sorry for remaining absent and not present with you in this symposium. Anyhow, I will present my lecture online. Our today's chairman sessions, Dr. Lingaraju, co-chairperson Dr. Santi, invited scientist Professor Gol Saab, Professor Walia Saab, Dr. Sharma, Dr. Bajaj, Dr. Sirohi, President of the Society, Dr. Pankaj, Head of the Division, Dr. Chawla, Project Coordinator, Dr. Khan, General Secretary of the Society, Dr. Abhijit, Nematologist from different parts of the country, entrepreneurs, students, ladies and gentlemen, and the members who are attending this symposium online. Now, before going to start my talk, I would like to thank D'Souza family for sponsoring this prestigious award. As well as I am also grateful to the members of the committee who identified me for this prestigious award. Friends, just now you had a Haiti. Now what next? Now you are going to test tobacco, and for that, let us go to BD Tobacco Research Station, Anand Agriculture University Anand, where we have worked and carried out our research projects. Now to know more about the BD, let us see what is tobacco. Tobacco, it's a commercial crop, cash crop, but different people and different organization, they consider according to their way. And if is to talk, the layman who is not non-user of tobacco, they consider tobacco as a useless, third class, addictive, and can't fill stomach. Society considered it is a blemish, disgraceful, anti-religious, and anti-social, while medical fraternity consider it as a health hazard and cancer causing. According to scientists, it's a miracle plant, and scientists consider tobacco as a guinea pig for research. As far as government is concerned, economically, it is a very important and next to petroleum product in income to the government. As far as employment is concerned, it provides employment to 45.7 million people in our country. How farmer consider tobacco? It is a golden commodity for farmers. In Gujarati, tobacco is known as tamaku, and farmer consider tamaku. Ta means tamaru means yours. Ma means maru means mine. Ku means kutumno kalyan karnar. means helper in family welfare. Means it is the commodity which help you, me, and our family. That is called tobacco. It is considered coined by the farmers. Now, as far as consumer is concerned, this consumer consider BD as a Swargani CD, ladder of heaven. We know with smoking we are reducing our life. That's why it is considered as a ladder of heaven. While the other products, the consumer consider it as a treasure of pleasure. Freedom fighter during Swadeshi movement, BD was considered as a Swadeshi against cigarette, and particularly during those times, Hind Mata was very famous product. Now about the area and economics of the tobacco, in India particularly, 0.27% of the net cultivated area that is under tobacco, that is 4.32 lakhs. As far as Gujarat is concerned, in Gujarat we have more than 1.5 lakh hectare under tobacco. Now economics is concerned, our country catches rupees more than 6,000 crores towards excise, rupees 55,000 crores, and export up to more than rupees 9,000 crores. As far as employment is concerned, tobacco provides employment to 45.7 million people, and which includes 6 million farmers and 20 million farm laborers. Now, as far as history is concerned, in short, tobacco cultivation was initiated in 6000 BC in America, but nobody knows about it. When Columbus landed in Tobacco Island, he noticed the local people of that island, they were smoking, chewing, and enjoying tobacco. So they and their team also tested it and tried, and they like it. So what happened? When they came back to Europe, they brought some seeds and plant material with them. And in the 17th century, that tobacco spreads into Asia and Africa. In India particularly, Portuguese introduced tobacco in 1605, and the first cultivation of tobacco was initiated in Gujarat, particularly Kheda and Mehsana district. Thereafter, it spread into the entire country. Now, tobacco was introduced in 1605, but until 1787, no research was carried out. But in 1787, when Calcutta Botanical Garden established, at that time, there they have started initial research work on tobacco, and that is about tobacco improvement. Now, today is a great day for me, for my crop, and our research. Because you see, right now we are conducting our national symposium in Calcutta. Then the initial research was initiated in Calcutta, that is the twin city of Howrah. And today, in the same symposium, our research and our team is awarded with this prestigious award. That's why it's a very important for us. Now, later on, what happened? From Howrah, that research was shifted to Pusa, Bihar, with the establishment of Imperial Institute. And from Pusa to Delhi, due to earthquake in Pusa, from Delhi, it was shifted to Guntur, establishing cigarette tobacco research station by IARI. And looking to the importance of tobacco, government thought it's giving very good commodity which can give income to us. So they imported excise duty on tobacco, and later on, government they also established Indian Central Tobacco Committee, and that committee and Government of India, they established Central Tobacco Research Institute in the year of 1947 at Rajamundry. And at the same time, in Gujarat, Institute of Agriculture, a private body, established BD Tobacco Research Station. And since then, both these institutes, they are working well. They have completed recently 75 years. And recently, CTRI has celebrated its platinum jubilee. So this is the history in short. Now, different types of tobacco, which is grown in India, that is cigarette and non-cigarette. In cigarette, FCV, non-FCV are there, and they are mostly confined to southern India. In Gujarat, we are working on BD, chewing, hookah, and rustica tobacco. Now, looking to the need of the farmer and trade demand, our center has developed different varieties. But out of all this variety, this GT9 MRGTH1, that was developed for the first time as a mosaic resistant variety from our station. Similarly, Anand BD Tobacco 10, which is found highly resistant to root knot nematode, and it is also developed at this station for the first time and released in the year of 2008 for cultivation among the farmers. Now, this tobacco also, just like other crops, attacked by number of pathogen, fungi, bacteria, flowering plant parasite, virus, and nematodes. to look after the nematodes. In the initial period, late Professor H.M. Shah Sahab started the work on nematode management in BD tobacco around 1960. Later on, Dr. D.J. Patel, we know very well, we all know him very well. He joined in 1966 and worked in tobacco up to 1986. Then he became professor. So on his vacant position, I was appointed to look after the nematological work of tobacco. Now, losses they have estimated, Professor Shah and Dr. Patel, under nursery condition, root knot nematode causing 51% damage, while in field crop, it is up to 50% loss they have estimated. Our team studied the impact of stunt nematode and reniform nematode, and they estimated 14% and 22% loss in nursery. Similarly, symptoms, these three nematodes, root knot, stunt, and reniform, they are a problem in BD tobacco field and nursery. So I will not go in detail of the symptoms, being we all are nematologists, but I will show you the pictures. This is the nursery which is infested with nematodes, and here you can see the seedlings of BD tobacco with gall roots. This is the close-up of nursery. You can see the growth and development. Similarly, in field crop, you see how plants are dying due to the nematodes, and these impair the quality and quantity of the tobacco. Now, this is the individual plant, and see the galling on the root system. This is it. Now, as far as reniform nematode is concerned, this is the treated bed and this is the untreated bed. You see the growth and development here also. When we uproot such seedlings, such type of growth is found. This is about the stunt nematode. You can see here sparsely developed root system where we inoculate a stunt nematode, while this is the healthy one. Now, this nematode, they are causing damage. Besides the other fungal pathogen, viral pathogen, they are also attacking. And when they unite together, they make havoc in the cultivation of tobacco. So here, let us see the symptoms, because many of you might have not seen tobacco crop, nursery or field crop, and the disease which they are causing. This is the frog eye spot. This is two viral diseases of tobacco, mosaic and leaf curl. This is frog eye spot under field condition. This is brown spot under field condition, and this is chlorotic mottle. Now, as far as management is concerned, in nursery, our team have worked on rubbing and soil solarization. Rubbing was found effective up to 40-45 days, while soil solarization remained effective till 60-70 days. And very good growth of seedling we are achieving here. And for this work on soil solarization, our team awarded Sardar Patel Agriculture Research Award in the year 2000 by Government of Gujarat. That is with citation and cash prize of rupees 1 lakh. Now, this is the rubbing view, how we did rubbing in nursery. This is the treated beds with rubbing, while this is the control bed. You can see the difference. Now, this is the solarized bed. This is the control bed, both sides, you can see. Now, here, this is the growth. See, this is in solarized bed, seedlings, and in control bed. This is the solarization on large scale in BD tobacco nursery on our farm. This is the result in the initial stage, and in later stage, you can see here the difference. Now, as far as management through cultural practices is concerned, in the initial stage, Professor Shah and Dr. Patel, they work and they come out with the conclusion that growing of winter crops, particularly chicory, lucerne, they tend to increase root knot disease and reduces the yield. They also tried castor anon 39-1, which is as a found trap crop in field crop, but ineffective in nursery, while this schedule found effective may be due to DBCP. This work was carried out by Professor Shah and Dr. D.J. Patel Shah. Now, they have also worked on organic amendment, and they come out with the conclusion up to 20 ton they found ineffective. But in our case, when we used 4 ton, not alone, but with castor or neem cake, followed by solarization, metalaxyl, and carbendazim, this schedule found effective and economical for management of nematodes, damping off, and frog eye spot. Similarly, Sunham green manuring also found effective and economical. This was carried out by our team. Now, so we have also worked on sowing dates. My team worked on this sowing date, and we come out with the conclusion. You see here, this is our normal date, which is regularly farmers they are doing. That is July 1st week. We extended this date up to July 3rd week, and particularly this first week is giving realization more than 3 lakhs rupees, while here we are getting more than 4 lakhs rupees. These 2 dates, they are not economical, effective. Now, Professor Shah and Dr. Patel also worked on biological control, and they found that these agents, Dictyria, Arthropatella, Dactylella, they are not effective in nursery and field condition. Similarly, ready-made formulation, nemin and natrin, was brought by GSFC, that is also found not effective in nursery. In nursery, they have also tried chemicals, particularly fumigant and non-fumigant. They were found very effective, but this insecticide was ineffective. This is the conclusion drawn by Professor Shah and Dr. Patel. Now, we have also worked, our team have also worked on Dazomate and carbosulfan. Carbosulfan found effective and economical, and it is recommended to nursery growers for management of nematodes. Dazomate found most effective in BD tobacco nursery, but could not remain economical. That's why it is not recommended. And for our work on Dazomate, our team awarded PP Single Memorial Pesticide India Award in the year of 1998 by Indian Society of Mycology and Plant Pathology. We have also worked on resistant and tolerant variety, and as an outcome, Dr. Patel, DJ Patel Saab, reported GT5 as a field tolerant variety in the year of 1986. But later on, our team worked on this, and we come out with the conclusion that Anand BD tobacco 10, which is found highly resistant to root knot nematodes in BD tobacco, and we released it in the year of 2008 for farmers. You see here, this is the seedling of ABT 10, this is A119, and this is the tolerant variety. We inoculated 5,000 nematodes. You see the root system at the time of harvest. This is the susceptible. This is the resistant variety. Now, my students also work on this variety, particularly resistant variety, and he studied the mechanism. You see here, these are the inoculated roots of ABT 10. This is of GT5 field tolerant variety, and this is A119 variety. And this is the control of the same variety. Now, this is the field view of Anand BD tobacco 10, which is highly resistant to root knot nematode. This is the individual plant. This is the single plant. This is again the field view. Now, this is the comparison of highly resistant variety with susceptible variety on our farm. See the clear-cut difference. This is the comparison on farmer's field on large scale. This farmer came to me, sir, my field is inspected, and next year we provided him 20,000 seedlings, and we told him, in the middle, you grow just 10 rows of susceptible variety, which you are going every year. So he did it, and you see the result. This 10 line completely failed, while this resistant variety survived. And this has increased our level of confidence to great level. And the farmer himself told me, sir, you are my God. If you have given me this variety before 5 years, I may be crorepati today. This is the under field, wherein we have observed during 7-8 we observed that root knot infested field is there. So we provided in 8-9 healthy resistant seedling to this farmer. You see the difference. Similarly, this is the individual plant of susceptible and resistant variety. Now, one of my students, she worked on mechanism of resistance, and she awarded with PR Verma MSc Student Award in the year of 2010. She came out with the conclusion: inhibitory effect of ABT10 on egg hatching, thick cortex of ABT2, high amount of nitrogen, phosphorus, sulfur, crude fiber, starch, and crude protein in roots of ABT10, significant increase in potassium and marginal reduction in magnesium in roots of ABT10 after nematode inoculation. Other advantages of ABT10 also we have worked out our team. See, our pathologists come out with the conclusion it is also moderately resistant to damping off. Then against leaf curl and frog eye spot, minimum disease was observed in this variety compared to other varieties. Now, in 2010, we got severe rain, that is more than 800 mm in 44 days out of 68 days nursery, and our entire nursery completely wiped out. But these two strips, which is of our resistant variety, they survived. This gave us clue that this variety is also resistant to wet foraging. Now, suckers, that is a severe problem in tobacco, and it's a headache and increasing input cost. But our variety, which is released as ABT10, resistant root knot, that is size suckering in habit, so less expenditure will be there on this variety. Now, as far as field is concerned, here also Professor Shah and Dr. Patel worked out that this summer crops increases the diseases, as this two cropping pattern, tobacco pearl millet, cotton fallow, 2 years propagator, another cropping pattern for tobacco pearl millet and castor and fallow, that is found effective. They also worked on organic amendments, Karan, Maruti, castor, groundnut, then leather mill, azola, but ineffective. and this Sunham green manuring, Sundia green manuring, that is also ineffective under field condition. Our team work on poultry manure and we come out with the conclusion at the rate of 45 kilogram nitrogen per hectare found effective and economical, and it is recommended to the farmers. Professor Shah also worked on fertilizers. They applied up to 600 kilo nitrogen, 180 kilogram from ammonium, anhydrous ammonia, but is ineffective. My team worked on date of planting in field condition also. In nursery, we have seen the result. This is the result in field condition. See, this is the September third week planting, and this is the August third week planting, susceptible variety. This is the difference. And we come out with the conclusion that you plant your tobacco in September third week. That is also managing the root knot nematode, leaf curl, and frog. So in summary, particularly in nursery, deep plow the nursery, avoid rabi or summer crops, do rubbing, soil solarization, or sow the seeds up to third week of September, or adopt adaptive seedling. If you don't want to do anything, go for resistant variety only. Now, these are the different IDM schedules that is released by our team. That is only for nematode damping off, and nematode damping off and frog eye. And that is also giving higher ICBR and good return. So it is recommended to the farmers. In field also, same like nursery, deep plowing, avoid summer crop, use poultry waste. Planting time should be in September 1st to 3rd week. Then use healthy seedling, use resistant variety, ABT10, and adopt crop sequences. Now, this is our research team. Professor B.N. Patel, who developed 2 mosaic resistant variety, and he was our head of the station as well as our department. Dr. Kiran Joshi is pathologist. He come out with the conclusion that ABT10 is having moderately resistant damping off and less leaf curl, etc. This Makwana, Delwadia, then Bhatt and PM Patel, they were, when SRS, Senior Research Assistant, they helped me in recording and observations as well as planning of our experiments. Then Rojasra, right now he is working on my position. Dr. Tulika Singh ji, she is assistant nematologist. She is in Department of Nematology, but voluntarily she helped us in planning of our experiments as well as in publications. Now my foundation bricks, my students who have increased my confidence to a great level. Arti Bairwa, she is right now nematologist in Potato Research Station, Simla. She studied mechanism of ABT10 against root knot nematode. And in PhD, she studied reniform nematode. Then Dr. Deepak, then Pradeep and Riddhi, they studied the population dynamics of nematode in BD tobacco field. Joshi worked on groundnut nematode. Right now, she is in IAS cadre in Gujarat government. Mr. B.K. Patel, he studied impact of soil solarization, rubbing, green manuring, and chemical on nematodes, seedlings, on physicochemical properties of soil and soil microbes. Then Ashok studied on stunt nematode. Devendra studied on pure leaf extract against nematodes. Then Poonam and Anjana, both are assistant professor. Poonam is at SK Nagar. She studied tobacco dust against nematode, while Anjana studied bulky manure, effect of bulky manure on nematode seedlings of tobacco and in soil microbes. Now, they are our backbones, our Honorable Vice Chancellor, Professor Sheikh Saab, Dr. N.C. Patel Saab, Dr. K.B. Kathiria Saab, and our Director of Research, Dr. Pathak Saab and M.K. Jala Saab. Earlier, Sheikh Saab and Kathiria Saab was also holding the charge of Director of Research. Without their encouragement, facility provided, and timely permission, we cannot achieve up to this stage. So for that, we are also grateful to him. Thank you very much. This is about my talk. Thank you. Now, any observation is there? I am there.
Ladies and gentlemen, we have come to the end of a very, very useful and practical lecture by Dr. Hemchandra Patel, recipient of Gerard C. D'Souza Memorial Award. And since it's an award lecture, questions are not entertained by the audience, members of the hall. And I invite our co-chairman, Dr. Shanti, to make any remarks. If any.
No remarks, sir. Thank you, sir, for your excellent information on BD tobacco. I take this opportunity to thank our organizer for giving me this opportunity. Thank you.
Thank you, Chair and Co-Chair.
Dr. Patel, we congratulate you for having completed his lecture right on the dot of 20th minute at the time given to us, to you. And I see that you respected everyone's time. Thank you very much and good night. I think that is still
Thank you very much, sir.
thick of the night. You are speaking to us in the thick of the night.
Thank you.
Sir, I take this opportunity to announce this award is been given by the society. There is a citation, there is a psalm. Since he is not absent, he is not here. So we'll try to send him along with a cash amount that will be given by check. So we'll try to send all the this award related material to his residence. Then this much we can.
Citation you can read.
Citation, yeah, you can read out this.
So the citation is given to for Dr. G.I.D. D'Souza Memorial Award, presented to Dr. Hemchandra Rajibhai Patel in recognition of his research contribution in the field of pneumatology in India at the National Symposium and the name of the symposium. Congratulations, sir.
Thank you for the opportunity. This particular award lecture concludes. Thank you.
Thank you very much, Chair and the Co-Chair. We are closing this session now, and we are starting the next session. May I request Dr. H.S. Kaur and Dr. A.K. Chobe. Dr. Kaur will be the chair of the next session, and Dr. Chobe will be the co-chair. We have three invited lead talks to be delivered by Professor S.B. Sharma. Professor Sharma will be delivering this online. Then Dr. Kaur will be speaking, and afterwards Dr. Walia delivers.
मेरा कंप्यूटर लैपटॉप लग जाएगा ये इसी काम इसमें नहीं लगा बस आपके ट्रांसफर
Good afternoon. To begin with this second plenary lecture session, I thank Dr. A.K. Chobe to join me as co-chair. There are three lectures. Dr. S.B. Sharma will be talking online on pneumatology in 2050, my vision. And followed by me, and then by Dr. R.K. Walia. I'm glad Professor Datta is here to listen to these lectures. So, in fact, without taking much time, I request Dr. Shashi Bhushan Sharma to give his lecture. In fact, Dr. Shashi Bhushan Sharma has been an illustrious pneumatologist, alumnus of III, and also he worked at ICRISAT and later on returned to IRA, Federal Division of Pneumatology, moved over to Australia, where he has done wonderful work in plant health and biosecurity areas, and he is recognized as number one pneumatologist in particularly West Asia. He has also been working on biosecurity aspects later on, and presently he has an NGO called Food for All, and also he is trying to promote particularly microorganisms, also microbial biodiversity for the benefit of agriculture, and also for human health. Without taking much time, I think I request Mr. Shashi Bhushan Sharma to start his lecture.
Yeah, thank you. Can I request Dr. Khan to share my slides, please, if possible?
Yeah.
In the meantime, he starts sharing my slides. I thank Professor Gaur for his very kind and generous words of introduction. And It is indeed my pleasure to be able to present this talk on a very topical issue. And I thought that first of all, I would like to congratulate the organizers, the local organizer committee, and also the national one for outstanding job they have done. I have a small sample size, and the book of abstract and the messages that I received regarding the arrangements being made, just outstanding. And I would like to congratulate them for this excellent work. At the same time, I would like to say hello to all the participants present here. It is more than, I think, 35 years that I have not been able to attend a nematology symposium in India. And I thought this time I will be able to, but due to certain circumstances. But I then thank the advanced technology that has enabled me to, if not physically, virtually contact you all. And this virtual contact is a great opportunity for me to at least see some of the faces I would like to say hello to. And it was good to see also Dr. H.R. Patel, with whom I worked in the late 80s. Can I have the next slide, please? No, before, yes. As I said that I'm speaking virtually, and this is where I am right now. Go to the next slide. This is where I am. This is Perth in Australia, and which is a beautiful city of Western Australia. It's the capital of Western Australia. Some of you may know Perth as from those who love cricket, that these are some of the fastest pitches of the world in cricket are in Perth. And Western Australia is known for two things, mining and agriculture. And Western Australia is also known for one of the nematological achievements, that is, to the best of my knowledge, Western Australia is the only region in the world where potato cyst nematode was found and successfully eradicated. And I have the proud privilege of being actively engaged in the project that led to the eradication of potato cyst nematode from Western Australia. As I said, when I received this invitation from Dr. M.R. Khan to speak, and then I accepted, but at the same time, can I go to the next slide? My memories went back to 1987. That was the time also we were talking about a vision for nematology for 2000. I vividly recall Dr. Dasgupta, a very well-known nematologist, our teacher, a visionary nematologist. He contacted me. I was at ICRISAT. He said that you got to give this talk on nematology in 2000 AD in dryland agriculture, in relation to dryland agriculture. I said, sir, I'm just busy with setting up nematology at ICRISAT, but he being my teacher, he said, you got to do it. And moreover, this is regarding the felicitation of your chairman, Dr. Gopal Sarup, you got to do it. So I had no option but to prepare this lecture and prepare this write-up. And it was difficult, but it was challenging. But then it also made me learn many things. From 1987 onwards, in the last 37 years, I've had opportunity to speak on vision on several other subjects and several other initiatives. And this is one thing which I learned. That learning was, can I go to the next slide, please? That whenever you see a future, it's a very complex process. It's a very complex process. You do not know what factors are going to impact on your future. Then is it based on what you have presently? The future is dependent on that. Or are there factors you are not able to see that will impact on the future? Or there are totally unknown factors which will impact on the future. And if you have to see those factors, it's extremely difficult. And the fourth was that you see that this is the type of future I would like to look at, and then you aim to get that future. Can I have the next slide, please? So what I'm going to do is I have looked at nematology in 2050. I have 4 scenarios. These are the scenarios I'm going to share with you. The scenarios for envisioning nematology in 2050. One scenario is based on the nematology currently as it is. So if that we take this one and then expand it further, how we think it will be in 2050. The other approach is that looking at nematology as a part of the bigger picture, which is agriculture and global economy, leadership within the country, globally and nationally, internationally, technology development. As part of that, how they influence nematology and how nematology would be in the coming years. Third is totally unforeseen variables. We don't know them at all. But what do we know? How do we envision things which we are not even able to perceive or see? So I have discussed that scenario as well. And the fourth and most important scenario would be that how we would like to see nematology in 2050. And can we, what we want to see and how we can achieve that scenario, how we can realize that scenario in 2050. So these are the 4 scenarios I would like to share with you in the coming 15 to 20 minutes. Can I have the next slide, please? Now, when I say that the based on present state of nematology, when we look at it, then it is perhaps not an extremely exciting scenario, present state. We look at the research, education, outreach, the support is diminishing. Number of nematologists engaged in teaching research, the number is going down. Plant nematology is not being taught in areas in schools or universities where it should be. Students are not ranking nematology as their first preference or something they would like to really aspire for getting into. And also the job opportunities are not very, very exciting. But then I had an immediate parallel that I realized when you go back and look at that in 1950s, people were asked in general that how do they envision life in year 2000? And whatever they envisioned, they were asked to write it, and it was stored. And 2000, when it was opened, then they realized that the majority of the people were very conservative about the future. Some of the people, they also predicted there will be Third World War. Now, then what happened? Then the assessment was that our vision for the future is very heavily impacted by our present circumstances. So when we look at those, and none of those who envisioned 2000 and 1950 did talk about that we will be landing on moon, we will be having a World Wide Web, we will have with mobiles, phones, and we will have the best of transport facilities. None of those. So perhaps we become very conservative if we are looking at from a very myopic point of view. So I did not pursue this further. Can I have the next slide please? But then there is another one. When we are looking at nematology in the 21st century, in a time when we have been very different to the agriculture in the 20th century. 20th century agriculture, we did an outstanding job. It was very much assisted by chemicals and many other inputs. But then when in 21st century, we realized that, look, we have fed about 6 billion people plus, and globally, our resources are increasing, our wealth is increasing, the technology advancement is unprecedented. And then we said that, look, we're talking about sustainability of the planet, sustainability of the food system, sustainability of almost everything with the resources that we have. When there's a very distinct change, a paradigm shift from in agriculture or food or the systems, where the first question the community is asking, is it sustainable? Is it causing any harm to our environment, planet, or our natural resources? Then all of a sudden, picture changes. So the soil has become extremely important as a resource, and its health is extremely important in terms of plants and human health. So when we look at not sustainability, sustainable agriculture, whenever we talk about it, so nematodes are an integral part of soil systems. So the interest on nematodes is per se given, is going to be much more than what it has been in the past. And then when we look at the technologies that we have had now, the ability that we have now to study nematodes, their impact, and the impact and how we can manage them, we feel very empowered. And then we have been in the past a bit conservative, and I would say defensive, in terms of losses caused by nematodes. Though we have a lot of good figures, but we always say that, look, they live in soil, they are subterranean, we cannot see them. Losses they cause are enormous, but it's very difficult to really articulate them. No, but it's possible that we will have techniques and also technologies that will allow us to very clearly indicate losses, qualitative, quantitative, in terms of soil health, plant health, planetary health, the nematodes are contributing to. So all this is put together. In the 21st century, where we have sustainability or regenerative agriculture or systems where soil and plant health is to be guarded, the input, the resources, the attention to nematology, in my opinion, is going to be much more than it has been in the past. Can I have another one? Next slide, please. Continuing the same thing, when we look at we have been working on plant parasitic nematodes, and we have a wealth of nematodes that are not plant parasitic, but they have a role in nutrient release, organic matter decomposition, and also in the soil health management. So whether we talk about ensuring agriculture becomes very sustainable, in all those role of nematodes across so-called free-living nematodes and plant parasitic nematode, it's believe is going to be much more getting attention than it has been in the past. So I would say that nematology is poised for a positive transformation. Only thing that is required is recognize and capitalize on the opportunities that we have right now. Can I have the next slide, please? And going ahead further, as I said, that in the 21st century agriculture, we are looking at biologicals, we are looking at microbiomes, we are looking at microbiomes, and we talk about that bulk of it, we say we have 1 trillion plus species, and 99.99% we don't know about them. They are in soil systems. When we start doing microbiome studies, and other soil organisms are going to be studied much more. And then we realize that nematodes, their beneficial impacts, we have studied, but not to the extent we have done for some of the other soil inhabitants. So the bioprotection research, the stimulus that we will have in the 21st century would also enhance our attention to nematodes, particularly the so-called free-living nematodes that you're talking about. Another thing which I also note when we look at the trends that we have, in the 20th century, we were very much pest focused in terms of management. But in the 21st century, there is a gradual shift towards a comprehensive approach, where we are managing plant and soil health, something like, the Dr. S.R. Patel talked about soil solarization. Soil solarization is something, it is not pest specific. Soil solarization, when you apply, whether it is a fungus, it's a nematode, insects, mites, they're managed by soil solarization. In addition to that, it has impact on positive microbes. And then once the polythene is lifted, our studies, and so back late in 1980s when we started this work on soil solarization, the positive microbes, they regain population, very quickly. So the positive impacts of soil solarization, they are much more, whereas the impact on the negative one, which take much longer to build. So in sum total, the strategies like soil solarization, which are broad and they're across pests and diseases, they're across other beneficial impacts, are going to be used much more. As a result, that nematode management would be more as a part of some other pest and disease management, rather than specifically looking for nematode management techniques or technologies. Can I have the next one, please? Now, this is something that we talk about when we believe the agriculture is going to be very much a sustainability build. We will have much more focus on soil and plant health. We will have biologicals, and all those scenarios indicate that we will have much more attention to nematodes, plant parasitic nematodes, as well as the so-called free-living nematodes, those who have a role in nutrient recycling and also soil health. But when we say unknowns one, we don't know them. For example, we have a new nematode species, which might bring, this very, very aggressive. It causes some of our key crops, which are staple foods, that are impacted to the extent we never even know. Or we find a nematode species which is so good for soil health that it is something like a lottery. So such changes could happen which we do not know. And then all of a sudden, then we realize that certain nematode pests may also impact, they are able to break the control option that we have created, that they are no longer controlled by those options. So all of a sudden, we have a new scenario. Now, when we talk about climate change, it has very many impacts shown, but impact on nematodes are not very clearly stated so far. It's possible that climate change impacts, when we look at that, we will find that nematodes may be more difficult to manage, or may become impacts on climate change when we understand them much more. Our approach to managing nematodes that are harmful may change, or our attention to nematodes that are very beneficial to us may change. So these are something unknown. But it's also possible that we may import a nematode species or any other population that causes significant losses to our crops that are very essential for our survival or for our commercial use. So all of a sudden, we are hit with a situation which we were not planning to. It's also possible that with the new technologies, tools, we are finding that we have a research which shows that nematodes are very integral in terms of relationship with microbes, and their presence is very fundamental to the soil health which we really cherish. So all those things are extremely important. Can I have the next slide, please? Next slide. Yeah. Then also it's something that's new technologies. It comes new technologies, tools. We always say that we can't see nematodes, we can't see their damage. That it's no longer their talk. We are able to see them. We are able to detect them. We are able to show there are some genetical. We find that nematodes have certain traits or parameters which we can use, or which we can use to manage them. There is also possibility that there are changes globally or national one, which impact on the way they say that, which become very favorable for work on nematodes. So there's also possible that as new crops are emerging, new food systems are emerging, that there will be new farming systems. And there could be new nematode problems or opportunities for us. But all those things indicate one thing, that we can use, or we can stand up to challenges or opportunities if we are prepared. So preparedness to meet the challenges, or preparedness to grab the opportunities, will be key to developing a nematology for future for 2050, when in the process that we are very well prepared. Can I have the next one, please? This scenario, the fourth one I'm talking about, is how we would like to see nematology. And that's extremely important. And I perhaps you would agree with me that we would like to see as a discipline which is a crucial discipline. It's essential for keeping plants and soil healthy at their best. We have identified key nematode challenges and opportunities, and farmers have easy and effective ways to manage them. Nematodes may remain out of sight, but definitely not out of mind of our researchers, policymakers, planners, politicians, anybody. So that's the future that I envisage, that 2050 we would like to have for nematology. But then if this is the case, then all of a sudden we have to look at how we go back to our current state of nematology. Current state of nematology is something which we have to see that we are nowhere near. we have to, if we continue to do what we have been doing all the time, then perhaps we will not be able to realize our vision for 2050 that we would like nematology to have. So there are very bold actions and decisions and changes are required. In fact, when I look at nematology globally, nationally as well, and one thing which I have to identify, I would say that nematology leadership has been assertive leadership has been not been very persistent. It has been there, but not very persistent. Our leadership in nematology worldwide has been very defensive leadership. We do a great job, but we do a very poor job in lobbying. And usually, why would a planner or a politician or a policymaker give attention to nematodes where we are not able to show symptoms clearly, not able to manage them very clearly, not able to provide management options very clearly? All of a sudden, this must also change. We must change. What we have to do is that now onwards, we have to take some bold decisions regarding leadership, regarding new initiatives. And can I have the next slide, please? And that's something which, this is the blueprint. I have written here that we have to do something where it's a national priority, national pride. And I would say that we have to look at eradication of ear-cochlear disease by 2035. This is one of the examples I have given here. Why do you do that? I believe this is doable. I believe that all the developed countries in this world have been able to eradicate ear-cochlear disease from their countries. And India right now is aiming, as the Prime Minister says, in coming years to the developed countries. So this resonates very well with the national perspective. So not only this eradication of ear-cochlear disease is not to be taken as a nematology initiative. It is to be taken as a national initiative. This is how we have to project it. And I have given 10 years term, around 10 years time, this is how much it takes to verify the eradication. But please consider this. Another one is eradication of potato cyst nematode.
So we have lost it. So you can hear us? Some network issue. You are muted, sir, somehow. So you are muted. You unmute yourself.
Just a minute. Yeah, can you hear me?
Now, yeah.
Can you hear me now?
Yes, it's okay now.
Okay. So let's go back to the slide of where we are talking about eradication of ear-cochlear disease.
This one?
Yes, thank you. I think I would even suggest nematologists, the Nematology Society, to take it as very seriously, because nematology have to have a national footprint. Nematology, the policymakers, planners must see that there is something being done which resonates very well with the national plans. We need to see what are the national plans, what we do in nematology links with those. And I would think some of the eradication plans, eradication of potato cyst nematode, it's important that we do a feasibility analysis. And then another one is root knot nematode. Root knot nematode is currently among the top 10 most widely spread pests in the world. And I believe my experience working in so many countries is that, are you able to hear me?
Yes, sir.
Okay. The root knot nematode is one of the most underrated pests. And this is this will be, in my opinion, will be among the top 10 pests in coming years. So definitely by 2050, this is in my opinion. The need for nematologists in India to commence a national project, something on the line of 1977 when we had SASIS project on international nematology project, but more so on the management. And the perspective is that we will reduce the root knot nematode cause losses by 10% in next 10 years. Something which resonates very well with the national plans. So this type of projects must be taken. The nematologists need to be much bolder and with assertive leadership in terms of projecting nematology in coming years to achieve our vision for nematology in 2050. Can I have the next slide? When I'm talking, next slide, please. Can you? Can you go to the next slide? So the next slide, I think, when it comes there, is primarily I'm talking about that as a priority. No, before this. Before this, yes. That we must take two actions, and that is we establish a nematology think tank. This nematology think tank, I will explain it further, and also establishing mentoring and coaching provision for nematology leaders. If I have to put in my resources, money, and bet for nematology to achieve its vision for 2050, we have to help the leaders. We help the leaders to be leading from the front, a very assertive leadership, so that they take the nematology, its perspective, to a new paradigm. For example, if we go to the next slide, when I talk about nematology think tank, I'm talking about 5 people, not many people, 5 very experienced people, nematology leaders, as permanent member. It's something like this nematology think tank is something like United Nations Security Council. It is these 5 members are there permanently. These 5 members are there to ensure that they are able to see the best for nematology and also able to assist and advise leaders in nematology to meet and to also realize the vision for nematology for 2050. Now, when I say 5 experienced members, what we are talking about is those who are visionaries, experienced, and they have the intellect, proven intellect. And their job primarily, this is not a leadership role. This is a rather invisible role. But this role is so critical to help the leadership deliver outcomes that are required for nematology to achieve its vision for 2050. And this is the think tank that will ensure that nematology remains dynamic, relevant, responsive, and as the most relevant discipline for the agriculture and environmental sustainability. So this nematology think tank is, in my opinion, extremely important for nematology society to consider. Can I have the next slide, please? And where I'm talking about is mentoring and coaching of nematology leaders. Nematology leaders, this coaching is extremely important because, there are nematologists who have retired from a job, but they have not retired from a profession. And immediately, immediately I remember Dr. Gaur, Dr. DJ Patel, and other people, those who are there. They have retired from a job, but not retired from a profession. The people like in every state we have people, experienced nematologists. We have to engage them. Their job is not over. They have to ensure that they're mentoring the current leaders or coming leaders to ensure that leadership is adequate to deliver outcomes that are required to achieve our vision for 2050, which we have defined. There is a difference between mentoring and coaching. Mentoring requires primarily the very experienced people, those who provide that mentoring. Coaching is skills-based. Coaching could be that to help leaders develop or further improve their skills for lobbying, for presentation, for this thing. So we need to identify mentoring and coaching provision for our leaders in coming times. And this is the role, I believe, is for Nematology Society, which must be taken seriously. Can I have the next slide, please? So I think what are the key actions needed? The talks like this one should not be very many. We should have more action plans. We should talk about just not the importance and all, to develop clear plans, actionable roadmaps, how we are going to be the most discipline, which is integral to agricultural productivity and for the sustainability of environment and for the economic growth of the country. And this is a country cannot do it alone. We got to develop international collaborations, education programs. We have to cultivate next generation of leaders. We have to have a pipeline of competent leaders coming out of those. So these are the actions needed. I think less talk, more action is something which we need to look at. Can I have that last slide, please? I think the potential for, as I say it, from whatever angle I look at, the potential for nematology in 2050 and beyond is immense. But it is not going to happen just because nematology is there. We got to make it work. And that will require actionable plans, concrete actions. You know, we may have all the tools, we may have all the technologies, policies, avenues, everything. But what will make it happen is effective leadership now and in the future. And that is where we have to work together to cultivate that leadership, to project that leadership, to support that leadership. One word that comes to my mind is, if you have three priorities, is leadership, leadership, leadership. And once we do that, I think nematology will be a discipline which we always cherish, that it should have its due place in agriculture and environmental sustainability. Can I have the next slide, please? Thank you very much.
I think, thank you, Dr. Sharma.
Yeah.
It's wonderful talk and good guidance about development of leadership, for concrete goals, action plans, and mentoring and coaching systems for leaders and leadership development. I believe every one of us, every individual, has got some leadership qualities. Even our students have got that. We need to nurture that. And of course, it goes with time, and they all will become certainly good leaders in their own domains. Somebody has to guide, and of course, it's a good idea to have a think tank. He has given a lot of thought-provoking ideas. I don't like to repeat here. Presentation is available. I will wish that every one of us sees the presentation, listens to that several times, and many new things will come up from this. I'm very glad that Dr. Sharma at this time he has given these ideas, and certainly it's not a gloomy future, I would say, as I see myself. He recalled that past when he came a little after me in this subject. When I came, it was even gloomier. When I was a student, I was taken out to the fields, and sometimes my own teachers used to say, "Why have you joined nematology? There's no future here, there, this and that." But I think I have seen a great future in nematology myself, and all of us have got that. So let us not be pessimistic. Let's be also optimistic. Things will come, and they'll keep on changing. New paradigms come, but we have to change with time. That is important. So thank you very much, Dr. Sharma. I think Dr. Sohaib also had something.
Thank you, Dr. Gaur.
Anybody from the audience has a quick question? We're running short of time, but any quick question anybody wants to say? Anyone?
Dr. Gaur, if you permit me, I'd like to say.
Yes, sure. Thank you. Please.
Can you hear me, Dr. Gaur?
Yes, please.
What I'm saying is that the choices we make or fail to make today will shape the direction of nematology in the years to come. So it's extremely important. We have all the raw material required. It's a matter of giving it a shape. And the conferences like this, so they are so important in coming together and bringing together all the ideas and delivering an outcome.
Fully agreed.
Thank you.
Yeah. Thank you.
Good afternoon. Second lecture of this session is to be delivered by Professor H.S. Kaur. And I think the need of introduction is self-explanatory. And the lecture is to be delivered by him is Nematology, Dawn and Memory Lane, the generational transformation. So I request Professor Kaur to deliver his talk on this.
Yeah. Okay, thank you. Good afternoon. I tried to save a few minutes for you, in fact, and try to be quick in my presentation. I'll be talking a little bit about the history part of nematology and how it has changed over time, over the past decades, where I've been observing. I've made this purposely these particular figures here, copied here, these cartoons. These problems of nematodes have increased, but not nematology. Nematology has remained down there in the bottom side. Nematode problems have increased, and this is where the opportunity for us comes, that we have to keep pace and we have to work harder and to make nematology also grow and to attend to these bigger problems now.
So you can change there.
No. There's no. But like a battery something. Okay. So if you see the recorded history about plant nematodes, particularly when you say plant nematology, although animal parasite nematodes or human parasites have been known even during the Vedic times, in all literature in different countries, whether it's China or Europe or India, it all mentions about animal parasite nematodes or human parasites. But plant nematodes, the first mentions we find in Shakespeare's writings, where he sowed cockle, reaped no corn. It means the effect of nematodes was known, particularly wheat ear cockle nematode. And then Needham in 1743, he saw these tiny nematodes coming out of these cockles, and this was called needle of cockle. This was sold, and this in fact was a discovery. This is not just a single-minded just to save nematodes, but basically to many things. It was about the non-spontaneous generation of life. Those are the times when people used to believe that life comes to spontaneously. We said, no, it's not involved spontaneous. It is something living inside here. It also story about cryptobiosis in nematodes. In nematodes, we know the survival strategies of nematodes are unique. And that's why they have survived this planet for billions of years before they came here, and they're still surviving in big diversity everywhere, all through the world, everywhere they're found. Cryptobiosis and anaerobiosis in animals, multicellular animals, were found with this discovery itself. Plant parasitic nematodes, of course, the first discovery as it directly. The foliar parasitic nematodes, unless they like they believe that nematodes are only in soil, but they are in soil, but they affect the above ground plant parts also. In the long-term survival, these nematodes can survive for many decades actually, in dry conditions and anaerobic conditions. Soil inhibitive nematodes, the first discovery of course was 1855, the root knot nematodes was found. 1859, the sugar beet cyst nematode in Germany, and 1913, the potato cyst nematode. These nematodes still continue to be serious problems. In spite of all kinds of words, chemical, physical, biological, cultural, integrated, everything we have tried, still they continue to be problems, and they are daunting problems actually. And the methods of the management of carbon disulfide onwards, we started doing that. Various kind of methods have been developed. That was the time when they started heating by steam or the chemical fumigants. Well, if you talk of India, pre-independence, the first report of root knot nematode in India, the plant parasitic nematode in India, was by C.A. Barber in 1901. He was not a nematologist. A botanist, in fact, working on sugarcane, did some work on pathology also. But then he found this nematode in tea, and later on, others found in other crops also. Ufra disease in 1913, Angona in 1919. Then 1930s, actually 20s and 30s, you find a lot of papers by Iyer and his team about the control of root knot nematodes in vegetables particularly. The white tip nematode in 1936, and the zoologists, they started describing nematodes now at various places in Hyderabad, in Aligarh, and other places, Kolkata also. They started describing nematodes, plant parasitic, free living, and even Jew parasitic nematodes, or they started describing. We see post-independence, it's a scenario of paucity of food, population started increasing, but food production was not increasing. And this was the time when our zoologists and botanists, they were studying nematodes, zoologists describing, botanists studying symptoms, teaching in botany classes that these nematodes also cause, particularly root knot nematode. And but this was the situation in the 1950s, when there were rampant food shortages. 50s and 60s, the shift to mouth existence. That was the time when any factor which was reducing crop yields by even a fraction was considered very serious. And that was the time when nematodes were found, particularly in wheat and potato. So the economic importance was given, the impact was felt by any of these factors, and the attention given by Sri Subramaniam and Swaminathan and many others, that paved the way. And that was the time when in 1957-58, the serious nematode of the Molière disease was discovered, and then the potato cyst nematode in Nilgiris in 1961. These two, besides the knowledge of the root knot nematode, Ufra and white tip and others, or the Yacogal, this paved the way. In fact, the government felt that these nematodes are causing so much damage to staple food crops also. And food security may not be possible unless we tackle these problems also. So that's why nematology gained importance. And the division of nematology was established in 1966, which became the focal center of the locus from where nematology started in the entire country, applied nematology as such. And this was also the division which later on, just 2 years later, started a postgraduate program of MSc and PhD in nematology, which is the number one in the world actually. The history is number one in the world. The nematology postgraduate teaching started at IRA or in India. Lot of training programs are there, Southeast Asia program, training programs, international training programs, with the help of UC Davis and also Wageningen and Rothamsted. A lot of people are coming and giving trainings. Like nematodes, movement of nematodes is slow, as you know. Nematology also remains slow. And still, hardly 9 or 10 universities in the country have nematology as a separate subject. The spread of nematology or the growth of nematology has remained rather stunted, I would say. It's mainly because many people don't really care about it, but there are other factors too. Well, during 1950s and 60s, you see, we moved from taxonomy. The generation change, you will say, fine. The traditional description of nematodes, whether it's Chambers or Amicov or India, Raspberry, I'm sorry to say that we have just recently lost Professor Raspberry, wonderful taxonomist, Bashir, or Siddiqui, and many others, Khan, so many others, B.M. Das and others, they were doing a lot of good taxonomy. Our description, microscopic descriptions, it was a challenging task. It was considered very important in those days. Later on, we moved to standing electron microscopy, and of course, the microscopy also we are looking into the private parts of nematodes, actually, to parenting patterns and all that. And trying to describe nematode species based on these small manual differences. Standing electron microscopy benefited more to understand this, give a greater clarity to the surface morphology and all. We moved to biochemistry as it started developing. We have the assays of phenotypes, proteins and assays phenotypes. They became important in differentiating species with other closely related species, which are difficult to distinguish by microscopy alone. We could confirm with the help of biochemistry. And now we have moved to the DNA using different kind of techniques of RAPD, RFLP, AFLP, and all that. We started moving, and then we are going to further into genome sequencing and other things. DNA barcodes have now become common. Dendrograms are made, barcodes are made. You can see this. You can see each individual species can be given a barcode. But this is possible only when you authentically identify whether by morphology or by molecular biology. You authentically identify a species, and then you give a barcode to it, and that becomes a reference kind of point. Long ago, in fact, Chawla is here. You remember around 2011, 2000 or so, when I was out of prison, I suggested to him a multi-layer photography of all these NNCI collection, and so that could be later on used as a reference rather than sending materials. And that could be, if they're ready, that could be used as a very good material for machine learning these days now. Machine learning and artificial intelligence we are talking, that could be the base for it. Unfortunately, still I think we are in infancy there. But that is the way we have to go. Well, we went to nematode management area. We started with chemical control, mainly DDA, DBCP, halogenated hydrocarbons, to demonstrate losses and also to control, in fact, as a method of control. But soon there, we understood that they have their own limitations. The large quantities they apply, non-availability in the country, and also the hazards, health hazards and environmental hazards. We understood that time itself. We moved on to carbamates, oxide carbamates. We moved on to organophosphates. And the story has gone on. Ultimately, we have these two nematicides now from Bayer and from Brahma. These nemates and Velbrine. In smaller quantity, they can be used. They are also toxic, but in small quantity, they are used, so the hazards are much lesser than that. This is the development there. But then good development here also has happened. The biocontrol agents and the botanicals. Neem and of course the Aspergillosis and Bacillomyces and others. We have a range of products. I'll just given a few here. When you go to the net, you'll find many of them. many products have now come up actually from these things, and we have products to say that the products are available in the market. So we have to give, we have extension activities can do that. In the field of ecology, also we transform from the area to area. We were working on life cycle and biology and molting and hatching and all those things, and their adaptation to survival. We are totally ready from, as you can see, the 3 layers of cuticle retained here, and only the parasitic stage comes only after that. And what is the role of this? This was they were helping in the survival of the nematode in the dying soil. And anaerobiasis and nematodes which are not cyst forming. That was also which was kind of discovered, and a lot of good work happened on that. We also worked on hatching behavior of nematodes. Hatching could change in response to environmental changes. Post-differentiate response that could be changed. We worked on chemotaxis, we worked on cryobiasis, chemotaxis, and some of these things are used in development of products now. But cryobiasis and anaerobiasis, they've been useful in the development of products in EPNs particularly. You can have dry products of these or even cold product or frozen products of these also can be available. We worked on, as I said, statistics particularly. We worked on models, correlations, regulations, partial regulations, other things. That was the time when I was doing nematology, in my graduation or postgraduate level. So that was the time when we were working on Janos models and others, correlations, regressions, and trying to correlate population density or yield, and assessing damage or to forecast damage. If this is going to the population, how much will be the loss? And that could be used as a basis of planning how much to invest for nematode management actually. We worked on distribution patterns and sampling technologies, sampling models. We worked on chemotaxis, particularly developed techniques and worked on chemotaxis, host, non-host, favorable chemicals, non-favorable chemicals, repellents, attractants, and all that. Things have changed now. We are going to signaling genes, gene activations, HSP genes, for example. So now the ecology also has changed towards more and wider ecological approaches have come into ecology also, and we are trying to understand the, now in fact, the soil biota itself can include study of genes, or study of extraction of DNA from the soil, and to know what kind of nematodes are there. And if you have the good database and barcodes, we can really find out, not by extracting by sieving and decanting, we can just buy extract sample from the soil, just like for microbes we do, you could also do for nematodes. There have been some major milestones in other areas also, from discovery of nematodes to discovery. I was just given a time scale here, from 1960 to 2020, you can see us here. We started from discovery of nematodes to description, classification of soil and plant nematodes, to microscopy, to molecular techniques. Just to summarize what I just said. Nematode anatomy also, to biology, to cell lineage. C. elegans, when I understood the cell lineage, and we understood that nematodes can become biological models. And when genome sequencing was done, it became a very good biotechnological tool. Survival strategies, and physiology and gene functions, pathogenicity, crop damage assessment, population and dynamics to forecasting. Right. Now, disease complexes, you are studying earlier only one fungus or one bacteria and one nematode, but now we are going to multiple interactions. All kind of being put together. Nematode control of physical, chemical, cultural, biological, chemical methods are there. We moved to integrated management, integrated nematode management, but we have now moved further into IPM. Nematode management has to be part of IPM. Shifts from, I already told about, from carbon sulfide to halogenated hydrocarbons, to oxygen carbamates, to organophosphates, to fluorobarium and sulfur, fluorosulfone. Ecology also, population dynamics to community structure, function, soil health, and climate by indicators. These two, particularly soil health, and we have been talking in the morning, nematodes are very important soil for in soil health also, besides playing other roles of plant parasitic or insect parasitic. The microbial and microbiological nematodes, they can be very, very useful in carbon mobilization and also about mineralization. Climate change bioindicators is an area which nematology still has to take up. Some work has been initiated abroad, not in India. The climate change, this is a bioindicator which can tell us to take a sample from an area. You can tell actually what kind of climatic change has happened. You take periodically or take different places. You can just compare the community structure of nematodes and you can find out what nematodes are actually, what nematodes are getting promoted, what are getting depleted, and that can be correlated very well to the climate change. So nematodes can be indicators of soil health and also indicators of climate change. So there have been shifts from applied to fundamental nematology. Many nematologists also went there, but unfortunately the number remained less. The number of nematologists remained limited. So most of them were taken away towards fundamental nematology. Applied nematology suffered actually. Well, we have plant resistance from screening that we spent a lot of years. For India Coordinated Project spent a lot of years. Actually, all centers doing a lot of screening work. A lot of lists of resistant materials are available, but hardly plant breeders are not taken any. Very few taken by plant breeders. This is where the support that had to come from other disciplines could not come to nematology. This is where we are support. We move to selection, we move to breeding, we move to transgenics, to gene editing, and of course we are not talking about CRISPR Cas. But the problem comes with the, although it is excellent technology, but there are scientific and sometimes pseudo-scientific or political reluctances and selfish trade interests that are preventing the adoption of this concept. Integrated nematode management, because now we have to talk of overall health, overall plant health, not only talk nematodes and insect or fungus or bacteria. We have to fit in somewhere here. When we say talk soil health, is many factors taken away, taken part in it, but we have to be fit in ourselves somewhere in the overall pest management scenario and develop specific modules that can fit in. Unfortunately, many of our techniques that we develop also manage other pest infections. So it's easy for us to fit in there easily, and not only for nematodes but for other sources. Entomopathy in nematodes, also the bacteria from the nematodes, their isolation, their mass culture, formulation of molecules from there. It is just possible that we can replace perhaps the BT gene or the chi proteins process that I was talking. We can have genes from Photorhabdus or genes from Leonorhabdus that can perhaps replace some of these BT genes and our dependence on other countries, Monsanto and others. Of course, we have to work on the risk to beneficial fauna also sometimes. So there is a good hope here, but also some need for caution. But again, a lot of people move from plant parasitic nematodes to entomopathic nematodes. Here again, there was loss to applied nematology. The number of nematologists could not increase, and that number has been just rather changing to other diseases. Whether it's good or bad is debatable, of course, it can be good or bad. EPN products also now available in the market, let's say many of them. Plant quarantine, particularly nematode quarantine, we have been practicing in the domestic and international levels also. But there have been some failures and some successes. We have prevented many nematodes from entering the country. But one nematode in which we had the domestic quarantine, potato cyst nematode, has now moved from the South India to the North India. And our potato growing areas of the north, they are now suffering from the potato cyst nematode. And this is where Professor Sharma talked that we have to go do something about it to contain or restrict or whatever, or to eradicate, I don't know, but it's a very big word to achieve. But at least we have to contain this. Education has changed from, of course, education training from classroom to virtual. This is generation change. We are now able to talk to the world, sitting anywhere. awareness and extension field visits and demonstrations, mass media, we have adopted here. More mass media is required, specific to nematodes and their knowledge to farmers. Still, the farmers remain, by and large, unaware of nematodes. There is a lot of awareness required, not only farmers, but also many others, our administrators, our scientists, and others. Global collaborations and networking have happened. From physical visits, now we are virtually able to interact with the world, and we are getting on. Nematodes as biological models, they have been used in genetics and biotechnology, environmental monitoring also, the bioindicators. In global climate change, there are some studies coming from Europe. We need a greater involvement of nematologists in collaboration of biotechnologists and environmentalists here. This is where again we have to extend our help. We have to go and tell them that yes, we can help you in understanding this and the techniques of this biotechnology and can help us. And also environmental scientists, they can find it as a good indicator in nematodes also. Genome sequences help that we know that what gene plays what function. So these developments collectively contribute to our understanding of nematodes, help in the development of sustainable strategies for managing nematodes, related issues in agriculture and other fields. This is now coming up from molecular angles also. Although we have made a lot of citable developments here, but have we lost? I say no. We have done a lot. We have developed techniques, we know nematodes, we know our problems. I say no, we have made successes and we have made achievements. But have we won? That's only partly. So a lot of things that remain actually to be attacked, and we have to develop our own working nematologists and leadership roles. We have to play, and we have to have greater challenges ahead of us. Shall we win ever? I will say never fully. The nematodes are very adaptive organisms. They are fast changing themselves. They adapt with nature. So we have to live with them. We cannot overcome them completely. So full victory may never come, but somewhere we may be near there. Okay? So failures in truly understanding nematodes and developing dependable management technologies are still lamented. So we have to still work on those. But we have to focus on some things and then attack it. So this is overall last slide, I think. The crop plus economic impact, development of social or the economic eco-friendly, economically viable integrated management modules to fit in the overall economy. This is where we have to go now. Reliable identification diagnostic tools we have to develop. We have to develop reliable and stable plant resistance through biotechnology or breeding or perhaps both. Safer and economical nematicides, effective biocontrol agents, techniques for mass production, formulation and application, understanding the role of nematodes in regulation and prediction of ecosystem function. This is where fundamental ecological measure. And then entomopathic nematodes for agriculture and human health programs also. We have to not only for agriculture, human health also, EPNs can work with. Present and future generations of nematologists, this is my vision actually, they will continue to convince the farmers, they will continue to convince the administrators, convince the policymakers, and even themselves. So nematodes are such organisms and their damage, the roles. Sometimes we ourselves start doubting what is happening. So we have to work ourselves, we have to convince ourselves and policymakers and scientists and others. The problem is even ARS and agriculture scientists, we talk to them, 90% do not know what nematodes are. We have to struggle ourselves, in fact, we have to struggle ourselves to have a UG program, UG course, at least 1 or 2 grade course in UG, so that every agriculture student knows that there is something called nematodes. The until food shortage is really, to starvation again, we have to start keep on, because when food shortage come again, they will ultimately realize this. But nematodes can cause food shortage. So food security is fragile. We should not say that we are now exporting, we are self-sufficient, we are all that. But this is just fragile. 1 or 2 monsoons fail, our food security will be somewhere else. Okay. So dear nematologists, do not give up your efforts. You will be needed always. Nematology still is an incomplete Taj Mahal. It looks beautiful, but some of these towers are still under construction. Nematology is still under construction. It will be beautiful and bountiful when completed. Thank you very much. My acknowledgement to I've taken many messages from the board. On the Gotia University, my colleagues have helped. All nematologist colleagues have taken the information, materials, techniques developed by them. All generations of nematologists. And also to Nematological Society of India and the Indian Statistical Institute for facilitating this presentation. Thank you very much.
Thank you, sir, for delivering the nice lecture, and you covered all the things related to the nematology. Pre and post independent India, and where we are, and what is the need of today's. So, is there any query or questions?
नहीं सर कंटिन्यू कर लीजिए
आपकी वाली है I'm sorry I took a few minutes more. It's because it's difficult to control when you are talking. Dr. Atewali, I think, is a wonderful nematologist, again alumnus of the Division of Nematology, one of the best students I have known and had, and also he led nematology at Risal in Anna, and also the coordinator of nematology at ASF Nematology. He has done wonderful work, and I'm sure many new ideas are going to come up from him. His work on Prasuria is still remembered. Thank you. We will be talking on the potato system network distribution, the policy analysis.
No, पहले वाला no previous one. No, please.
No, not this one. No.
Am I audible? Yeah. Talking of potato cyst nematodes, I'm reminded of Dr. Seshadri, who was the driving force regarding PCN in Nilgiris. He was my teacher, my advisor. He used to speak for hours together on PCN because he had the first-hand field experience. Dr. God had also been my teacher, but he was very young at that time. Dr. Seshadri teaching theory classes and Dr. God taking practical classes.
Still young.
He says he's still young. It was a deadly combination. I'll quote one small incident. He used to take our practical classes, but he was very keen to know our performance in theory examinations. What was your score in the midterm? We had never obtained so many marks. I said very proudly, 24 out of 25, sir. He was not amused. Only 24? What is this? I was surprised, but I didn't say anything. I could not say. But there was a message for all these students over here. Why you lost 1 mark? You got 24, but why you lost that 1 mark? That was the message. That was the inspiration of Dr. Seshadri, Dr. God, that I still cherish. Anyway, let's come to the topic, potato cyst nematodes. You will understand why I put a question mark here, policy paralysis. Shift me all right?
You change it from there.
Definitely. Okay. I have organized my presentation. I'll start with the global dissemination of PCN. Then briefly I'll talk about the scenario in Nilgiris deliberately for the benefit of students. But my main concern is about the nematode PCN in North India, how it disseminated, what is the current scenario. So regarding global dissemination, as we know, most of these things you know already. It originated from South America, not moving. That is animation. Yes. See, it originated from Portugal and Peru to Europe, and Europe became the secondary dissemination center. From rest of the world, it was actually from dissemination from Europe, including India. So this is how it disseminated throughout the world. And for India in particular, started from Peru to Europe and UK particularly, it was transmitted to India. That is what we believe about 200 years ago. Now the present scene in the Nilgiri is, it was of course intercepted in 1961 by Dr. Jones in Nilgiris, and there were some more interceptions during 1964-65. But based upon this discovery and our experiences in Europe and USA, the Tamil Nadu government, they started one project, Golden Nematode Scheme, in which the field-to-field surveys were conducted for PCN. Those who have visited this area may be knowing that when we go up to Nilgiris, start the ascent, there is a town, Mathiaplayam, on the foothills, from where all the produce comes to Mathiaplayam, and from there it is passed on further. So the surveys and all these things were conducted in Mathiaplayam, field go-downs and other things, to trace how it is going and where it is going. And the most important thing, which most of you know, the introduction of domestic quarantine in 1971. And then came the Indo-German Nilgiris Development Project. I'll particularly highlight these things. And initially, of course, it was in Nilgiris, the containment, but later on it was realized that the adjacent areas of Kerala and Karnataka also, they had this infection. Regarding this Indo-German Nilgiri Development Project, the German government provided desalination, free of cost. And it was a different kind of approach. The approach was to eradicate PCN by using very high dosages, 30 kg AI per hectare in the first year, 15 kg, the third year 7.5 kg, like that. That was the purpose, and huge quantity, you can see in that table, huge quantities of desalination was pumped in Nilgiris. And there were other provisions also in that domestic quarantine. The produce from Nilgiris was not allowed to move out of Nilgiris, and whatever produce was there, it was to be used only as stable potato, not as seed potato. But over the years, the potato production has gone down drastically in Nilgiris. You will see at present it is even less than 2,000 hectares. That is true even now. And the main reasons for this is the late blight of potato, that is the serious problem. Potato cyst nematode is also a serious problem. And some of these areas is replaced by tea gardens, tea plantations, and carrot. Farmers have started substituted potato with carrots. It's a different matter that carrot is now again confronted with root knot problem. That is a different issue. Anyway, after this, regarding this species, both these species, they are present simultaneously. Out of 5 pathotypes of Rostokiensis, RO1, RO2, RO5 are present in Nilgiris, and Pallida, all 3 biotypes are present, but Pallida is dominating. 57% populations, they are Pallida, and 43% are Rostokiensis, and the major means of dissemination is the soil which is adhering to the tubers. That is the carrier of cysts worldwide. And one more thing you must try to understand, most of the nematodes will die without water, including cysts of Heterodera. If you keep them without water, the eggs inside will be killed. This is one nematode which you keep without water for many years. This nematode, eggs inside this nematode cyst can still survive. So that is the main carrier, the soil adhering to the tubers. And even in a normal looking crop, 15% losses are very common. And in a heavily infected field, you may not harvest even what you have used for sow. Now, the current measures they use in Nilgiris is besides the domestic quarantine, production is still in position. Crop rotation with non-solanaceous vegetables, mainly it is French beans and carrots. Carbophrone application is still going on in split dose, one at the time of sowing and the second dose at the time of earthing up. But CPRI has developed nematode-resistant varieties. There are 3 varieties I have listed here. Started with Kufri Swarna during 1980s or so, Kufri Nilima 90s, and recently Kufri Sayadri. These are the nematode-resistant varieties, and these are quite popular in that area. And CPRI has also standardized the protocol so that the nematode cysts do not disseminate with these seed tubers, to treat these seed tubers with 2% sodium hypochlorite. That will kill the cysts, whatever is there with the seed potato. So there are dedicated centers in Ooty, CPRS, Central Potato Research Station, and TNU Regional Station. They are all dealing with this particular problem. Now, I come to the main topic. What is the situation in North India? Our mindset was, till recently, this nematode is confined to about 4,000 hectares in Nilgiris and surrounding areas. That's it. Nobody thought beyond that. But there was a suspicion it may be present in North. And in 2010, first time it was published on record that this particular nematode is present in Shimla. Not only in Shimla, it is present in the farms of Central Potato Research Institute. Kufri, you may be knowing, it is just a few kilometers from Shimla, where most of the potato varieties are developed, because this area is virus free. So all the varietal development activities conducted at Kufri, Kyo, like that. There are 2-3 more farms. After this report, we thought in AICRP, if it is present here, and this is a premier center from where germplasm exchange takes place throughout the country, why it is not present in Uttarakhand? Why it cannot be present in Jammu Kashmir? Agroclimatically, these are similar areas. So we started our surveys on priority, and our surveys conducted during 2011 to 2019. We published it. This paper I am citing here, most of what I speak is already there in this paper, Current Science. Prevalence and spread of cyst nematode Globodera in northern hilly areas of India. Now I'll talk about these 3 states. What is the situation in Himachal, then Uttarakhand, then Jammu Kashmir. In case of Himachal Pradesh, there are GPSFM, this means Government Potato Seed Multiplication Farms. Throughout Himachal Pradesh, besides CPRI, there are many government potato seed production farms. Germplasm exchange takes place in between these, among these, all these. So we focus mainly on these seed production farms. Out of 11, in 7 districts, 9 were found heavily infested with this nematode. Only that Lahore Spiti area and Kinnaur area, their samples did not reveal. And one more thing, wherever we recorded the presence of this nematode, the height altitude was more than around 2000 meters and above generally. And in low altitude areas, we could not encounter this nematode, or if at all we encountered, they were mainly empty. Empty means they might have been introduced, but the nematode failed to establish. Uttarakhand. One more thing I want to add here, all these potato farms, government potato farms that I'm referring to, these are very old farms, established by mainly Britishers, and many of them are in inaccessible areas. Even now you cannot go with motor, you have to walk down a few kilometers. Okay, so these farms in Pithoragarh, I have highlighted here, Balati, Tixen farm and Oli farm, very heavy populations of cyst nematodes. And other stories more or less same, altitude more than 2000 meters, lower than that you don't get. Farmers field we also was surveyed, but in farmers field the intensity and the incidence is not to that extent. And then in the Jammu and Kashmir, Nathata, you must have heard that resort, famous resort, Patni Top. It is close to that. That is the first time we recorded in Jammu. Jammu and Kashmir has two divisions, Jammu division and Valley. In Jammu division, these are the places where I highlighted, these are all government seed production centers. And in Valley, in fact, we could not travel much. In Shopia, we encountered, this is the picture of farm manager at Natha Top. I pulled out the first plant. When I showed him, he became nervous. Sir, please don't tell it to my director. I lose my job. Why should you lose your job? It goes to your credit. You have discovered a cause why the yields in your farm were going down over the years. So you will be appreciated. Don't worry. I'll help you. So like that, we demonstrated them. And again in Shopia, you will see this is Kashmir Valley. They were not at all familiar with this nematode. Nobody knew it. You pull out any plant, you get plenty of cysts. This is how we demonstrated them. And overall, if I summarize, in these three states, Himachal, Uttarakhand, Jammu Kashmir, these are the main government seed production farms where you can see they are all above 2000 meters height, altitude. Frequency, last column you see, more or less 100% in all these farms, and heavy cyst population per 100 cc soil. That is the situation. And you please concentrate on this particular graphic. This is quite interesting. You'll see the color in the background is pink and yellow and bluish and purple. Purple is the lowest height. And the lowest height you will not find incidence of the nematode. Mostly it is in the yellow and blue areas. And the dots in different colors, that is the population of cysts. Red, more than 300 cysts, 200 cc soil. Brown, yellow, green, and black. I draw your attention to see these particular things. So this is Shimla. You have very high populations, moderate, still lower, and least. So these black dots are mostly the farmers' fields. And this yellow and these things are mostly the state seed production farms. I have, I think, enlarged these things. In the next slide, you will see here. Rajouri. You will see those yellow circles and black dots. Yellow is the seed production farm. And from seed production farm, usually the adjoining farmers, they are dependent upon seed production farm for quality seed. They prefer to collect it from them. Then in Himachal Pradesh, again, this is the focus, and in Uttarakhand also, these are the empty cysts with cross, means the nematode was introduced, but it could not establish, it failed to establish because of temperature, altitude, and other factors. So what I conclude from this is, from institutional farms, say Central Potato Research Institute, it goes to the regional stations. I'm talking about germplasm exchange. Regional stations, government seed product multiplication farms, from there the farmers fields. I've taken this text from the website of CPRI, where This germplasm, this seed is distributed to 15 units in the country at our regional stations. And the breeder seed is supplied to State Department of Agriculture and Horticulture, means the seed production farms. This is how we could establish that this is the scenario and this is the vicious circle, how it goes on. And the story is yet to be completed. This is just a tip of the iceberg, I say. It is there in Himachal, must have gone to Uttarakhand and Jammu Kashmir, but focus on those blue states. There are high altitude areas there. There are government stations there. CPRI station is also there in Shillong. We need to survey all these areas. Even other states also, these 3 states also, this story is incomplete. So we need to do a lot of surveys. So one more opinion developed that because it was introduced in Nilgiris, it was restricted here, and because of germplasm exchange, it has gone to the north. That is the opinion which we developed. But let me compare the north and the south India situations. In south, it is uniformly distributed across the 4,000 hectare areas, including farmers' fields. Very high populations. Go to any field. North, it is mainly in the high populations are mainly in the government seed production farms, and the incidence and intensity is low in farmers' field. So that is the comparative situation. So what we recommended is whether it has gone from south to north. Let us compare the homologies of the population of cyst nematode from south versus north. That will unravel what it is. What is the similarity or dissimilarity? I'm sharing the data from Dr. Sombanshi's lab. It is still to be published with his permission. All these populations from Himachal, Uttarakhand, Jammu Kashmir, you see the altitudes, and they have done molecular analysis of all these populations. So what is the conclusion from these molecular studies is these populations are genetically similar. All these populations in North India, they are genetically similar, and they most the Indian populations, they have high similarity with the Bolivian populations. And it has mainly, the another conclusion is it has come from this South America, Bolivia or Peru, these are adjacent countries. So that is the origin. But what we lack here is the comparison with South India populations. So only 50% purpose is achieved, not the full purpose. Okay. One more publication I'm referring to you, which I read only 2-3 days ago. This is a publication by CPRI scientists, Seed Potato Production Systems in India. Chronologically, they have cited the events. I came across a reference. First time in India, potato was introduced in 17th century by Portuguese people, and that was used in Karnataka and Surat in 1675. Even if it is infected with nematodes, it will not establish in Surat and Karnataka plains. That is convincing. Another reference is, it was introduced in Shimla Hills 1828 and Nilgiri Hills 1830. We all know 1830 Dr. Shishadri used to refer, but this information was new to me also. In Shimla, that means in Shimla and in Nilgiris, it was introduced simultaneously. So that means these two things are happening independent of each other, but simultaneously. One more interesting thing. Much before the establishment of CPRI, that was around 1956, Central Potato Research Institute, there were government potato breeding institute in Shimla since 1935. And it had 2 seed production farms. One is in Bawali Hills. This should be read as Kumaon slopes in Uttar Pradesh. Now it is in Uttarakhand. And Kufri. So these 3 farms, Potato Breeding Institute Shimla, Kufri and Kumaon, these 3 were already existing, and these 3 units were merged around 1956 to form Central Potato Research Institute, what is now Central Potato Research Institute. So what I mean to say is, this might have happened 18 something when it was introduced in North India, in these particular farms, government farms, before the formation of CPRI. Again, if you read this article, there are multiple references of introduction of improved potato varieties at different timings. So it's a complex situation. So like that I have highlighted here, Himachal Pradesh developed a research scheme to produce disease-free tubers up to date, which was imported from Northern Ireland. Like that, there were many importations. So that is the current situation. Now, problem is highlighted. What is the solution? We presented our case to the ministry, Joint Secretary Plant Protection, sir. He was Dr. Rajendran, IS officer, but he was PhD in agriculture from IRI, not in plant protection. So he understood our case very critically, my presentation. So I explained to him all these international development, domestic developments, and future likely what is to be likely to happen, all those things. Even at that time, some officers from all these three states, CPRI, were invited in that meeting. All of them were still not taking it seriously. When I narrated this thing, then the chairman, Dr. Rajendran, he said, if I agree it is not a serious problem today, it has to become potential, it has the potential to become a serious problem tomorrow. So let us devise measures today. So we made recommendations, short-term measures. This was aimed to fumigate the entire area, farms, by the government itself. CPRI and other farms, government farms, they should have tuber washing facilities so that the cysts are dislodged before the produce moves out. And the nematology departments to help, and other, the government issued advisories to these three states to follow some measures which are listed here, midterm measures. The CPRI was to provide seed of Kufri Swarna, which is a resistant variety, you know that, and training to all the officers, horticultural officers, and one more very important thing that we suggested them, like potato seed production farms, they have farms for the production of other vegetables also, seed production of other vegetables also. So what we suggested, why don't you swap this? The other vegetable farms you convert into potato farms, and other vegetables you can bring here in potato farms for at least a decade, so that the nematode population declines.
Natural crop protection.
Yeah, this is what is there in the recommendation. In the long term, we have to test. We were supposed to test the new chemicals and continue surveys to map out areas. And the best thing that happened because of this is the domestic quarantine that was restricted to Tamil Nadu at that time was extended to Uttarakhand, Jammu Kashmir, Himachal also. But there was a problem in this. If you implement it blanket, there are some aeroponic units, seed production units in Himachal Pradesh, where they produce quality seed, which is given to the plain areas. Means with this restriction, they could not move it. So they approached ministry, and then this was modified. Corrigendum issued. Only those farms where we had intercepted this, recorded this, so these restrictions were imposed only in these farms. So we conducted training of the officers in Jammu, in Uttarakhand. Dr. Satya Kumar is here, Dr. Rajan Salalia is here. So we were part of the team. We worked together, and Dr. Chandel in Himachal Pradesh. And the new chemicals were also tested. I think this is the story about 5 years back. I need to update it. The other new chemicals must have been tested. But I have a feedback that these chemicals, they give very good results in quality and quantity of production. And in conclusion, I will say, this seems to be quite widespread in North India also, but the heavy populations we get in PCN farms only, that is because they go for monocropping, potato after potato after potato. And it is mostly above 2000 meters altitude, and the eastern states. There we need to focus. We have not sampled those areas, and this needs verification. Some more molecular studies are required to derive homologies between South India populations and North India populations. And mainly till today, whatever data we have, I would say it is mainly the government seed production farms which are playing the major role in the dissemination of the nematodes. But it is all happening because of ignorance. So this is all the work which we did under AICRP in Himachal Pradesh, Dr. Chandel, Jammu Kashmir, Dr. Rajan Salalia, Uttarakhand, Dr. Satya Kumar. If they have anything to update this information, we are free to do so. And I'll conclude by saying this is the forthcoming book in which we have 3 chapters on potato alone. South India, the quarantine paralysis by young nematologists. North India, wake up call by an old retired nematologist, and root node nematode, also on potato. So this book is about to be published. Thank you very much. I thank you much more.
Thank you, Dr. Walia, for a wonderful lecture on a very important problem actually, and we are all concerned about it. This also indicates one important thing. What I said, what about the ignorance of many among the scientists also, besides the farmers, about the nematodes? And not only ignorance, if some of them knew, they were underestimated. So underestimation and ignorance both have contributed to this. This is now spreading and will continue to spread. Only one caution as an ecologist I can say that we should not be complacent just by saying that they are found only in upper hills or more than 2000 meters. Even if cysts are found or empty cysts are found in the lower hills, it is possible that in due course, maybe after 100, 200 generations, nematodes are very adaptive organisms. You will find races, you will find populations, they will adapt to warmer areas also. Also, I see one more thing. If you see the map of India, there are many areas of Punjab and other places also where the same latitude as these parts in Himachal Pradesh. And there are also cooler areas in these states also. Maybe adjoining parts in states also we have to be vigilant. I'm not saying it's there, but we have to be vigilant about this. So not that we wake up only when we find problems. So we should be vigilant. And the surveillance program, the national program, as Dr. Sharma mentioned, at least if not eradication program, it is national surveillance program and restriction program has to be there on this important, widely important problem. Thank you.
Thank you.
Can I ask something, Dr. Gaur? Can I ask something, Dr. Walia? Can you hear me?
Yes, please.
Can you hear me, Dr. Walia?
Before you ask me, I have to request you to share. You mentioned about the eradication program in Australia. Okay. So can you share your experiences with us, with this audience? Because the Indian scenario is quite different. What is your experience of eradication of PCN in Australia, please? And then you please put your question also.
Dr. Walia, thank you very much. It's very old, Walia. For a question, there is a question. So no, that's very good. Look, I did mention about the PCN eradication in Western Australia. And I would also mention as my role as Director of Plant Biosecurity in the state of Western Australia, which enabled me to sit on eradication of more than, say, 500 incidents across pests and diseases. So what I'm saying is not because of any emotions. It's a lot of experience in dealing with eradication programs. And the eradication program in Western Australia took 25 years. And here, as you know, that biosecurity is if you ask a taxi driver, he will give you a lecture on biosecurity. So that's the common public knowledge about biosecurity and the need for actions to be taken. So there was, for example, the detection of a cyst led to a very stringent regulatory enforcement from day one. And that they remained there for 24 hours. I was looking at your slides that washing of potatoes or allowing table potatoes to be given. Here, no such things. For example, from day one. So the scenario in Western Australia is very different because it was found only in one region, one location, where different places. So we could then trap it, and ultimately it took 25 years still. But the Indian situation, for example, when I would look at it, there are certain questions I would like to see that of the total potato area, in your opinion, what is the percentage that has PCN infestation? That's one. And secondly, has there been any consideration given to assess the feasibility of eradication? The reason why I say is when we look at the eradication programs here or any other parts of the world, then sometimes we look at it, it's not technically feasible, and the end of story. But if you look at for potato cyst nematode present there and spreading, so what is the damage it's going to cause in 500 years? So there is no point talking about 1 year or 10 years. So when we talk about 100 years or 500 years, and then you make an assessment, the amount of money, resources that we're going to spend versus what we are going to spend right now. And the need for, the reason I was talking about the creating visibility and linking nematology initiative with the national initiatives was from this reason.
Okay. See, as far as I know, the major potato growing areas that fall in plains in India, only 5% potato is grown in hilly areas. And out of those hilly areas, we, in case of Nilgiris, they sampled all those hilly areas in joining Nilgiris, and it was restricted to around 4,000 hectares. And the effort was made to eradicate nematodes by pumping in decinate ice, as I said, 30 kg per hectare, but that effort failed. There are provisions, there are quarantine provisions, there are other advisories issued to North Indian states also. The problem lies with the implementation. Whatever advisories I listed, whatever recommendations we made here, I was today morning asking Satya Kumar in Uttarakhand, what has happened after that, after 5 years of our recommendations. They say that those farm managers, now they are rotating within that farm, 1/4 area this year, next year 1/4 area, 1/4 area, 1/4 area, like that. Imagine the amount of rainfall that happens in hilly areas. It is pouring, and the amount of soil that is drained down with the water, that is definitely bound to carry cyst. It is fortunate that the nematode does not establish in plains. Otherwise, it would have been a havoc. These are the things are in place. Implementation part is the problem. That is my judgment. I may be wrong.
If Dr. Gaur, if you may allow me, I think it's an important topic. What I would say is that implementation will continue to be a problem as long as it is seen as a nematology problem. The moment it is seen as a national problem, national priorities, implementation will not be a problem. That's a fundamental difference as to eradicating a nematode or a pest is science-based. Eradicating a pest or disease is also art of using science to deliver an outcome. So that is where we have to ensure that important nematode like potato cyst nematode should be given at least a go for looking at the feasibility of achieving eradication. Is it possible? That feasibility study is important. And I would request a person like yourself, esteemed nematologist, Dr. Walia, consider doing it.
No, we are very thankful to you for your comments, and we try to do our, as nematologists, we also feel concerned. Who would not like to see the eradication of PCN in India? We would also like to see that. Now, again, once again, the practical things, the ground level realities are very different. That is it. Who will check
is producing Nilgiri heels is used for table potato or it is going out as seed potato. There is no check on this. Scientists cannot. Government
डिस्कस करने लग जाएगा क्वेश्चन.
Anyway, we'll consider your suggestions and your recommendations. We will give a serious thought on this.
It's a very plausible suggestion, in fact, actually, and we should at least make a kind of point of recommendation from this particular symposium. This is a problem to be taken seriously. And also, if possible, we should go for other academic programs. But until then, we should at least start restricting it as far as possible. And because the problem here in India, farmer is free to grow anything. There is no control actually. There is no regulation which can stop a farmer from growing anything. They bring seeds from anywhere. There is no restriction on where he gets the seeds. All that. So there has to be larger kind of restriction program, regulation. A very good act has come out, but this has to be implemented properly. And the district administration and others, they have to be sensitized. And in those areas, there can be a special kind of meeting with those agricultural officers of the state or something. We can perhaps, including ICI scientists and university scientists, we can put together, we can have a joint kind of meeting and emphasize that this has to be taken up seriously. And at least, if you're not 100% dedicated, at least can be restricted to a great extent. And alternative crops have to be provided. It's like carrots there here also alternative crops have to be put in. Demand side itself may not be a solution, but alternative crops have to be. We cannot ask the farmers to take it fallow, whatever.
Sir, I have one question. Actually, now the seed transport is restricted, not restricted, totally banned from 3 states. So the director of CPRI has been writing to the ministry for relaxation on that. They say that there is a technology to completely remove this.
Sodium hypochlorite, I mentioned.
Sodium hypochlorite. So with this technology available,
CPRI can do it, but how many other farms will do it? That is the problem.
that is what. Those who can do it, should they be allowed for seed selling? That's one question, because they have put that letter to me.
Okay.
And it has to be discussed along with the this quarantine.
I'll quote again Dr. Sharma's suggestion. I was hearing his presentation. Nematologists have to be bold, aggressive, not in a denial mode. In this particular case, we have been in a denial mode. For 10 years at least, it was kept under the carpet. Don't reveal it.
Yes.
It has happened like that.
And my second question is, you yourself have observed that it only occurs about 2,000 meters or feet, whatever. So now, if the seed is sent to the plains, and we know that it was happening since several decades, and it has not established in the plains. What are your suggestions if they are allowed to transport it to the plains?
Immediately there may not be problem, but in the long run, as Dr. God also mentioned, these partially embedded cysts may also get adapted in the long run under plain conditions. So the new biotypes may develop, but this is not going to happen immediately.
Yeah, but how sure we are that it is going to happen even after 100 years? Because almost 100 years have gone since we know that the potato cyst nematode has been here. So is it wise to restrict the transport of seeds to the plains?
So after treatment, you can send it, no problem. After treatment, washing facility, sodium hypochlorite treatment, there is no problem. After that, you can send it. Okay, and there are preferences between these two species also. Pallida likes lower altitudes. Rostockiensis is more prevalent in higher altitudes. Okay, so maybe with the introduction into plains, Pallida slowly over the decades may develop a freak population that may establish.
The thing is that we still have 100 more years for us to develop a technology to manage it. Isn't it?
As of today,
So,
whatever technology is available with us.
Thinking of the risk 500 years from now.
See, that is a
appears to be
natural
too
phenomenon.
much.
That is a natural phenomenon that happens not only with this nematode, that can happen with any other nematodes also. That is how the nature survives. Okay. Whatever we have, the best possible means today is cleaning, treatment, go.
So you think that
I recommend it. I recommend it.
Yeah, you recommend it.
Yes.
So if the discussion comes, I can put this.
You
point
can
there.
put it.
No, just something. There are two things here. One has to be sure that the sodium hypochlorite treatment will give 100% removal of disease.
So they claim that with this treatment, these fish are killed, destroyed totally.
So you have to be sure that it's 100% control. Secondly, there is an alternative available. It's a aeroponically grown potato seed. Why can't that be used as a seed rather than this?
You cannot produce that much of seed, and it is
It's
costly also.
it's a small facility required. Which can be
We can promote it. We can promote it on a large scale.
From Pago Farm itself, they were selling about 40 to 45 lakh rupees of seeds, CPRI only.
No, with,
there are many other farms
with
who were
huge
selling
production and more scaling up with the production, the cost and the availability will increase.
If you have aeroponically grown potato seed, you can have RS-free potatoes, you can have bacteria-free potatoes, you can have fungal-free potatoes, you can have nematode-free potatoes. Why not, if you have a technology, why not go for that? Why not to just give up this type of technology of using mass the tubers as a seed? Alternative technology available.
Dr. Gaur, sir.
It may not be 100% effect. So no control is 100% effect.
Actually, sir, they use 2% for 30 minutes. 30 minutes is too long a time, and 2% is too high a concentration. So that appears to be okay, but I am not sure. That can be.
No, I've read that paper. It's exactly like that.
Dr. Gaur, may I make a suggestion, Dr. Dr. Gaur?
There should be some education program also then, that this has been really treated with their way.
Dr. Gaur, can I make a suggestion here?
Yes, sir.
Yeah, Dr. Gaur, I think Gautam, what he is saying is that we have to be also clear about the role of nematologists, also regulatory control, is to enable businesses rather be restrictive. So my submission would be for Dr. Walia, I think it's Dr. Walia's project, to consider a scientific panel which is looking at 2% sodium hypochlorite treatment, and scientific panel approves that this is a treatment which provides the level of control that we require. Then the regulators endorse it, and it goes to the traders that they can use it. So there is a formal way of doing it rather than a recommendation from a technical panel, which has not gone to a regulatory panel.
Very good. Yes, if you can.
Yeah, my question relates to the effectiveness of the treatment, tumor treatment that's being done. Yes, these microclouds of, let us say, aggregates of soil adhering to the tumors carrying cyst is one story, and the tumor treatment has to be done. Right. We remember distinctly our venerable past hematologist, Dr. Yasheshal, included, telling that, okay, tumor treatment is fine, we are rid of cysts. But what about the sacs? What about the granny bags? See, if you actually, you are required to treat them. That's what my thing. Okay, 2% hypochlorite, whether it's more efficacious or not, let's debate it later. But the sacs, what is being done to the sacs, the bags? Because they contain hell lot of
They also
cysts.
contain cysts. It was reported from Madhya Pradesh, my report.
Correct, but are they being treated? they should be treated as well, and they should be, if they are not in a, if we are not in a position to treat them, we should destroy them. Doesn't matter. This is my question, sir. This is my concern or question. Thank you.
You want to ask something? No, I am not. Mute.
Someone? There are no more questions. I think we can conclude this session.
Thank you. This talk generated a lot of interest. Thank you very much.
I also thank my co-chairs.
Thank you, sir. Thank you, chair and the co-chair for. So I all know that there is I don't know what's the time now.
Very nearly 3 o'clock.
So we'll break for lunch. So and the lunch would be definitely for 1 hour. But within this 1 hour, whoever presenting the poster, please be aware that the judges would adjudicate your poster will be visiting at some point. Possibly in the second half an hour of the lunch break. So I'm assuming it would be, we are breaking from 2:50. So 2:53, then 3:20. So 2:50 to 3:50 would be lunch break, and then 3:20 onwards the judges will be adjudicating your posters. So please stand by them. We will convene sharply, quote unquote, at 3:50. So let's hope for the best for the rest of the day. Thank you. So one more thing is that during the lunch break time, I would shout out and try to get the group done today. So everything is now hot.
To get our presentations as soon as possible. I think it's fine. ये लैपटॉप तुम ले रहे हो, मेरा वाला मैं दे रहा हूँ इधर, लगा देना। उसको यहीं छोड़ दे, मैं बैग को। ये सुदर्शन ना बैग रखे के लिए खुद ही है?
ना, कोई खुद ही नहीं।
आपका